Beneath the sun-scorched plains of southeastern Romania, hidden under 40 feet of rock and clay, there exists a world that has never known daylight. A world sealed away from the surface for 5.5 million years. A world where life does not depend on the sun, where creatures have evolved in total isolation, where the very chemistry of survival has been rewritten from the ground up. This is Movile Cave, one of the most extraordinary and least understood ecosystems on planet Earth. It is a place where the rules we thought governed all living things simply do not apply. It is a place that challenges everything we believe about what life needs, what life can endure, and what life can become when left entirely alone in the dark. This is the story of a sealed world, a forgotten biosphere, and the astonishing creatures that call it home. Please let us know in the comments where you are watching this documentary and what time you are watching it. And be sure to subscribe to the channel for future videos. To understand Movile Cave, we must first understand how profoundly alien it is compared to everything we know about life on the surface. We live in a world dominated by sunlight. Every green leaf, every blade of grass, every towering forest and sprawling meadow operates on a single fundamental principle: photosynthesis. Plants capture the energy of the sun and convert it into sugars. Those sugars feed herbivores. Herbivores feed carnivores, carnivores feed scavengers. And so the great chain of life continues link by link, all of it ultimately tethered to that burning star 93 million miles away. Remove the sun and in most places on Earth, life collapses. The food web unravels. The energy source disappears. Without photosynthesis, there is no primary production. Without primary production, there is no food. Without food, there is no life. This is the rule. This is the paradigm that has governed biological thinking for centuries, and Movile Cave breaks it completely. Here, in a space no larger than a modest house, life thrives in absolute darkness. Not just microbial life, not just a thin film of bacteria clinging to wet rock, but an entire ecosystem. Spiders, leeches, water scorpions, crustaceans, worms. Dozens of species, many of them found nowhere else on the planet, living and breeding and hunting and dying in a place that has not seen a single photon of sunlight in over 5 million years. How is this possible? How can a complex ecosystem sustain itself without the sun? The answer lies in a process called chemosynthesis, a form of energy production so fundamentally different from photosynthesis that it seems almost like science fiction, and yet it is real. It is happening right now beneath the plains of Constanta County, in a cave that most people have never heard of. This is a story about darkness and endurance, about isolation and adaptation, about the extraordinary stubbornness of life in the face of conditions that should, by every reasonable measure, be lethal. And it is a story that may hold the key to one of the greatest questions humanity has ever asked: are we alone in the universe? Let us begin at the beginning. Let us go underground. The story of Movile Cave's discovery is in many ways as remarkable as the cave itself. For millions of years, this underground chamber existed in perfect obscurity, sealed beneath layers of limestone, clay, and less fine-grained sediment deposited by ancient winds. The surface above it gave no hint of what lay below. There were no sinkholes, no natural entrances, no springs bubbling up from the depths to betray the presence of a hidden world. The cave was, for all practical purposes, invisible. Its discovery in 1986 was entirely accidental. Romanian authorities were surveying potential sites for a new power plant along the Black Sea coast, near the town of Mangalia. As part of the geological assessment, workers drilled test boreholes into the ground to evaluate the stability of the underlying rock. One of those boreholes punched through into an unexpected void, a cavity in the limestone that should not have been there according to the existing geological maps. A Romanian scientist named Cristian Lascu was among the first to investigate. Lascu was a speleologist, a cave researcher, and he recognized immediately that this was no ordinary geological feature. The air rising from the borehole was warm and humid, and it carried a sharp, unmistakable odor, the smell of hydrogen sulfide, the same gas that gives rotten eggs their distinctive stench. This was unusual. Most caves in the region were dry, cool, and well ventilated. This one was clearly different. Lascu and his colleagues expanded the borehole and carefully descended into the darkness. What they found defied every expectation. The cave was small, just a few hundred meters of passages and chambers, but it was alive. The walls were coated in thick, spongy mats of microorganisms. The water in the lower chambers was warm and reeked of sulfur. And moving through this hostile, lightless environment were creatures, pale, eyeless, ghostly animals that had clearly been living here for a very long time. The scientific world took notice almost immediately, but access to the cave was severely restricted from the start. The Romanian government, recognizing the extraordinary nature of the discovery, limited entry to protect the fragile ecosystem within. To this day, Movile Cave remains one of the least visited places on Earth. Fewer than 100 people have ever entered it. By comparison, more people have walked on the surface of the moon. The cave is sealed with a heavy concrete cap, and entry requires special permission, extensive safety equipment, and a willingness to crawl through narrow, sulfurous passages in conditions that can only be described as punishing. This extreme restriction is not bureaucratic excess. It is absolutely necessary. The ecosystem inside Movile Cave is so delicate, so finely balanced, and so utterly unique that even a small disturbance, a change in air composition, an introduction of foreign microorganisms, a shift in temperature could have catastrophic consequences. Every person who enters the cave brings with them billions of bacteria on their skin, in their breath, on their clothing. These foreign organisms could potentially outcompete the native microbial communities that form the foundation of the cave's food web. A single careless visit could unravel millions of years of evolution, and so the cave remains locked away, studied carefully and sparingly, its secrets yielded only to those patient and disciplined enough to approach it on its own terms. To appreciate what makes Movile Cave so extraordinary, we need to understand its geology, the physical structure that created and maintained its isolation for millions of years. The cave is located in the Dobrogea region of southeastern Romania, a flat, semi-arid landscape near the western shore of the Black Sea. This region has a complex geological history stretching back hundreds of millions of years, shaped by the collision of tectonic plates, the advance and retreat of ancient seas, and the slow, patient work of water dissolving rock. The bedrock beneath the Dobrogea plain is Sarmatian limestone, a type of sedimentary rock laid down approximately 11 to 13 million years ago, during the late Miocene epoch, when much of this region was covered by a shallow, warm sea. This limestone is composed of the accumulated shells and skeletons of countless marine organisms, foraminifera, mollusks, corals, and other creatures whose calcium carbonate remains settled on the sea floor and were gradually compressed into solid rock. Over millions of years, as the sea retreated and the land rose, this limestone was exposed to weathering and erosion. Groundwater, slightly acidified by dissolved carbon dioxide, seeped through cracks and fissures in the rock, slowly dissolving the calcium carbonate and creating voids, small at first, then gradually larger. This is the process of karstification, and it is responsible for the creation of caves, sinkholes, and underground rivers throughout the world's limestone regions. Movile Cave was formed by this process, but with a critical difference. Unlike most karst caves, which maintain some connection to the surface through sinkholes, fractures, or permeable rock layers, Movile Cave became sealed. Approximately 5.5 million years ago, during the Messinian salinity crisis, a dramatic period in Mediterranean geological history, when the Mediterranean Sea partially or completely dried up, conditions in the Dobrogea region changed dramatically. Layers of impermeable clay and less accumulated above the cave, cutting off its connection to the atmosphere and the surface ecosystem above. This seal was remarkably effective. Isotopic analysis of the cave's atmosphere and water has shown that the air inside has a composition radically different from the surface atmosphere. Oxygen levels are roughly 1/3 to 1/2 of normal surface concentrations, between 7 and 10%, compared to the 21% we breathe on the surface. Carbon dioxide levels are enormously elevated, reaching 100 times the surface concentration in some measurements. Methane is present in significant quantities. And hydrogen sulfide, that toxic, foul-smelling gas, permeates the lower chambers at concentrations that would be dangerous or lethal to most surface organisms. The water in the cave tells a similar story of isolation. Geochemical analysis has shown that the thermal waters rising into Movile Cave originate from deep underground, from aquifers far below the limestone, where water has been heated by the Earth's internal warmth and has dissolved minerals and gases from the surrounding rock over geological timescales. These waters are rich in hydrogen sulfide, methane, and ammonium, compounds that would be toxic to most surface life, but that serve as the energy currency for the cave's chemosynthetic ecosystem. Radiocarbon dating and other isotopic studies have confirmed that there is essentially no exchange of organic matter between the cave and the surface. No leaves fall in, no insects wander down, no rainwater seeps through carrying dissolved nutrients from the soil above. The cave is, for all biological purposes, a closed system, a world unto itself, operating on its own internal energy budget, independent of the sun and the surface biosphere. This degree of isolation is almost unprecedented. There are other caves on Earth that harbor unusual organisms or extreme environments, but very few that have been completely sealed from the surface for millions of years. Movile Cave is not just a dark place underground. It is a separate world, a biosphere within a biosphere, and it has been running its own evolutionary experiment for longer than the genus Homo has existed. The atmosphere inside Movile Cave is, by any surface standard, hostile. Imagine descending through the narrow artificial shaft that provides the only human access to the cave. The air grows warmer as you descend. The cave maintains a constant temperature of about 21 degrees Celsius, roughly 70 degrees Fahrenheit, significantly warmer than most caves at similar latitudes and depths, which typically hover around 10 to 12 degrees. This warmth comes from the geothermal waters below, which act as a slow, steady heater for the enclosed space. The humidity is extreme, close to 100% at all times. The air is so saturated with moisture that condensation forms on every surface, including your equipment, your clothing, and your skin. Visibility is limited not by the absence of light, that is absolute, but by the fog-like conditions created by this pervasive humidity. And then there is the smell. Hydrogen sulfide assaults the nostrils almost immediately, a sharp, acrid, overwhelming stench that triggers an instinctive revulsion. This is not surprising. Hydrogen sulfide is toxic to most aerobic organisms, including humans. At low concentrations, it causes eye irritation, headaches, and nausea. At higher concentrations, it can cause pulmonary edema, unconsciousness, and death. The concentrations in Movile Cave's lower chambers are not immediately lethal to a healthy adult with brief exposure, but they are far above what would be considered safe for prolonged habitation. Researchers who enter the cave wear gas monitors and limit their time in the most sulfide-rich areas. The oxygen deprivation adds another layer of difficulty. At 7 to 10% oxygen, the atmosphere in Movile Cave is comparable to high-altitude conditions far above the summit of Mount Everest. Human visitors experience shortness of breath, dizziness, impaired judgment, and fatigue. Physical exertion becomes difficult, cognitive function declines. Every movement must be deliberate, every action carefully planned because the environment offers no margin for error. The carbon dioxide levels compound the problem. High concentrations of carbon dioxide cause a condition known as hypercapnia, accelerated breathing, confusion, and eventually loss of consciousness. Combined with the low oxygen, the elevated carbon dioxide makes the cave's atmosphere genuinely dangerous for humans. Researchers must monitor their exposure carefully and retreat to the surface at the first sign of physiological distress. And yet, in this atmosphere that would kill a human in hours, life flourishes. The organisms of Movile Cave have adapted over millions of years to conditions that we find intolerable. They breathe this thin, poisonous air as naturally as we breathe the clean, oxygen-rich atmosphere of the surface. They swim in water laced with hydrogen sulfide. They feed on microorganisms that derive their energy from the very chemicals that would kill us. They are, in every meaningful sense, aliens not from another planet, but from a parallel world that exists beneath our feet, operating by rules so different from our own that they challenge our most basic assumptions about what it means to be alive. The foundation of the Movile Cave ecosystem is chemosynthesis, a word that sounds technical and abstract but describes a process of breathtaking elegance and importance. To understand chemosynthesis, we must first revisit photosynthesis because chemosynthesis is its dark mirror, its underground counterpart, its proof that life has more than one way to power itself. Photosynthesis, at its most basic, is the conversion of light energy into chemical energy. Plants, algae, and certain bacteria capture photons from sunlight and use that energy to split water molecules, releasing oxygen and combining the hydrogen with carbon dioxide to form sugars, organic molecules that store energy in their chemical bonds. These sugars are the fuel that powers the plant's own metabolism, and when the plant is eaten by an animal, that stored energy passes up the food chain. Every salad you have ever eaten, every steak, every piece of bread, all of it traces its energy back to photons captured by chlorophyll. Chemosynthesis is fundamentally different. Instead of using light energy to drive chemical reactions, chemosynthetic organisms use the energy released by chemical reactions themselves. Specifically, they oxidize inorganic compounds, substances like hydrogen sulfide, methane, ammonium, and ferrous iron, and use the energy released by these oxidation reactions to fix carbon dioxide into organic molecules. The end result is the same organic compounds that can serve as food, but the energy source is entirely different. No light is needed. No sun is required. All that is necessary is a supply of reduced inorganic chemicals and an oxidant, typically oxygen, though some chemosynthetic pathways can use other electron acceptors. In Movile Cave, the primary chemosynthetic process involves the oxidation of hydrogen sulfide. Specialized bacteria, chemoautotrophs, take in hydrogen sulfide from the thermal waters and react it with the limited oxygen available in the cave atmosphere. This reaction releases energy, which the bacteria use to convert carbon dioxide into organic carbon, the building blocks of life. These bacteria are the primary producers of the Movile Cave ecosystem, playing the same role that plants play on the surface. They are the foundation of the food web, the organisms that create organic matter from inorganic ingredients, the bridge between the mineral world and the living world. But hydrogen sulfide oxidation is not the only chemosynthetic pathway operating in Movile Cave. Methane oxidation is also significant. Methanotrophic bacteria consume the methane that rises from the deep thermal waters, using it as both an energy source and a carbon source. These methanotrophs represent an alternative pathway of primary production, adding to the total organic carbon available to the ecosystem. Additionally, nitrifying bacteria oxidize ammonium, another compound present in the cave's thermal waters, to nitrite and then to nitrate, releasing energy in the process. While nitrification is a less energetically productive process than sulfide or methane oxidation, it contributes to the overall chemical cycling within the cave and provides nutrients that other organisms can use. The result is a multi-pathway chemosynthetic system, with several different types of bacteria exploiting several different chemical energy sources, all operating simultaneously in the dark. This redundancy is important. It provides the ecosystem with a degree of resilience. If one chemical energy source fluctuates, others can compensate. It also allows for niche differentiation, different bacterial species specializing in different chemical reactions, reducing competition and allowing more species to coexist. The total energy budget of the Movile Cave ecosystem is, by surface standards, modest. The rate of primary production, the amount of organic carbon generated per unit time, is far lower than in most surface ecosystems. A temperate forest might produce 1,000 grams of organic carbon per square meter. A tropical coral reef might produce several thousand. Movile Cave likely produces a tiny fraction of this, perhaps a few grams per square meter per year, though precise measurements are difficult given the restricted access and the technical challenges of working in the cave's hostile environment. But this modest energy budget has been sufficient to sustain a complex ecosystem for millions of years. The organisms of Movile Cave have adapted to life in an energy-poor environment. They are smaller, slower, and longer-lived than their surface relatives. Their metabolic rates are lower. Their reproductive rates are lower. Everything operates at a reduced tempo, a slower rhythm of life dictated by the limited energy available from chemosynthesis. This is a profound lesson. It tells us that complex ecosystems do not require abundant energy. They do not require sunlight. They do not require the prodigious productivity of a tropical rainforest or a productive ocean upwelling zone. They require only a steady, reliable source of energy, however modest, and enough time for evolution to build complexity from simplicity. Movile Cave has had both, and the result is one of the most remarkable biological communities on Earth. The microbial communities of Movile Cave are not evenly distributed throughout the cave. They concentrate in specific locations, forming structures that are visually striking and biologically crucial, the microbial mats. These mats are thick, spongy, floating, or wall-attached accumulations of bacteria and other microorganisms that represent the densest concentrations of life in the cave. The most prominent microbial mats are found at the air-water interface in the cave's flooded chambers. Here, where the sulfide-rich thermal waters meet the oxygen-containing atmosphere, conditions are ideal for chemosynthetic bacteria. The bacteria form dense, intertwined communities, some species oxidizing hydrogen sulfide, others consuming methane, still others processing ammonium or other reduced compounds. These mats can be several centimeters thick and cover significant areas of the water surface, creating a living carpet that is the primary food source for the cave's animal inhabitants. The microbial mats are not homogeneous. They have a complex internal structure, with different microbial communities occupying different zones based on the local chemical gradients. At the mat's surface, where oxygen is more available, aerobic chemosynthetic bacteria dominate, organisms that require oxygen to oxidize their chemical substrates. Deeper in the mat, where oxygen has been consumed by the surface layers, anaerobic organisms take over bacteria that can oxidize chemicals using alternative electron acceptors like sulfate or nitrate. This stratification creates a series of microenvironments within each mat, each with its own chemical conditions and its own specialized microbial community. The result is a remarkably diverse microbial ecosystem packed into a very small space, a miniature world within a miniature world, with its own internal complexity and its own internal food web. Molecular studies of the microbial mats have revealed an extraordinary diversity of bacterial and archaeal species. Using techniques such as ribosomal RNA gene sequencing, researchers have identified representatives of numerous major bacterial phyla, including Protobacteria, Firmicutes, Actinobacteria, Bacteroidetes, and Chloroflexi, among others. Many of these sequences represent previously unknown species, organisms that have never been found anywhere else on Earth and that may have evolved in isolation within the cave for millions of years. The archaea are particularly interesting. Archaea are a domain of single-celled organisms that are distinct from both bacteria and eukaryotes, and they are often associated with extreme environments, hot springs, hypersaline lakes, deep-sea hydrothermal vents. In Movile Cave, methanogenic and methanotrophic archaea play important roles in the carbon cycle, producing and consuming methane in a complex interplay that helps regulate the cave's atmospheric composition. The microbial mats also harbor a range of eukaryotic microorganisms, single-celled organisms with complex cells, including various protists, fungi, and microalgae. The presence of fungi is particularly noteworthy, as fungi play important roles as decomposers in most ecosystems, breaking down dead organic matter and recycling nutrients. In Movile Cave, fungi likely perform a similar function, helping to decompose the remains of dead bacteria and animals and making those nutrients available for reuse. Together, these microbial communities form the base of the Movile Cave food web. Everything else in the cave, every leech, every spider, every wat or scorpion, every crustacean depends directly or indirectly on the organic carbon produced by these microorganisms. The microbial mats are the cave's fields and forests, its pastures and plankton blooms. They are where the energy enters the biological system, where inorganic chemistry becomes organic life, where the deep Earth's chemical wealth is converted into the currency of biology. Above the microbial foundation, the Movile Cave ecosystem supports a surprisingly diverse community of invertebrate animals. Over 50 species have been identified in the cave, spanning multiple phyla and representing a range of ecological roles: grazers, predators, scavengers, and parasites. Of these 50-plus species, approximately 35 are endemic, found only in Movile Cave and nowhere else on Earth. This level of endemism is extraordinary and reflects the cave's long isolation and the unique evolutionary pressures it has imposed on its inhabitants. The animal community can be broadly divided into two zones based on their habitat within the cave: the terrestrial zone and the aquatic zone. The terrestrial zone includes the dry or damp passage floors, walls, and ceilings above the water level. The aquatic zone includes the flooded chambers and pools where the thermal waters accumulate. In the aquatic zone, the most prominent animals are various species of crustaceans. These include several species of amphipods, small, shrimp-like organisms that are common in freshwater and marine environments worldwide, but that have evolved into unique species within Movile Cave. The cave's amphipods are pale, eyeless, and significantly different from their surface relatives in both morphology and behavior. They feed on the microbial mats and on detritus, the accumulated dead organic matter that settles on the cave floor. Also present in the aquatic zone are isopods, another group of crustaceans, related to the familiar pill bugs or roly-polies found under logs and rocks on the surface. The cave's isopods, like its amphipods, are depigmented and blind, having lost these features over millions of years of life in total darkness. They are bottom dwellers, crawling along the submerged surfaces and feeding on microbial films and organic debris. Copepods, tiny crustaceans typically less than a millimeter in length, are also found in the cave's waters. These minute animals are important components of aquatic food webs worldwide, serving as food for larger organisms and as consumers of bacteria and single-celled algae. In Movile Cave, copepods likely fill a similar intermediate role, consuming bacteria and being consumed in turn by larger predators. One of the most remarkable aquatic inhabitants is a species of leech, Hemopes kika, the blind leech. This animal is a predator, feeding on other invertebrates in the cave's waters. It is completely eyeless and unpigmented, with an elongated, flexible body adapted for navigating the narrow crevices and flooded passages of its underground home. The blind leech is a powerful symbol of the cave's evolutionary processes, a familiar type of organism, transformed almost beyond recognition by millions of years of adaptation to a world without light. The aquatic zone also harbors a water scorpion, Nepa anophthalma, another eyeless species found only in Movile Cave. Water scorpions are predatory insects, related to the more familiar water bugs found in ponds and streams on the surface. They breathe through a long, snorkel-like tube that extends from the rear of their body to the water's surface, allowing them to obtain oxygen from the air while remaining submerged. In Movile Cave, where the air above the water is low in oxygen and high in hydrogen sulfide and carbon dioxide, this breathing strategy requires special adaptations to cope with the toxic gases. In the terrestrial zone, the animal community is equally fascinating. Several species of spiders have been found in the cave, including species of the genus Agricina that are endemic to Movile Cave. These spiders are, like their aquatic counterparts, pale and eyeless. They spin webs in the cave's dry passages, catching small flying or crawling invertebrates. The presence of web-building spiders in a cave ecosystem is remarkable. It implies a sufficient supply of prey organisms to make web construction energetically worthwhile, which in turn implies a level of ecosystem productivity that was initially surprising to researchers. Pseudoscorpions, tiny, scorpion-like arachnids without stinging tails, are also present in the terrestrial zone. These diminutive predators hunt even smaller invertebrates, using their oversized pincers to capture and subdue prey. Like the spiders, the cave pseudoscorpions are blind and unpigmented, having evolved in complete darkness. Various species of springtails, tiny, six-legged arthropods that are among the most abundant animals on Earth, inhabit the terrestrial zone as well. Springtails are important decomposers and fungivores in surface ecosystems, and they likely play similar roles in Movile Cave, feeding on microbial films and fungal hyphae that grow on the cave's walls and floors. Beetles, millipedes, and mites round out the terrestrial fauna, each species showing the characteristic traits of cave adaptation: reduced or absent eyes, loss of pigmentation, elongated appendages, and enhanced sensory organs for detecting chemical and vibrational cues in the dark. The diversity of this community is what makes Movile Cave truly exceptional. This is not a cave with a few bacterial films and a handful of simple organisms. It is a complete ecosystem with multiple trophic levels: primary producers, primary consumers, secondary consumers, and even tertiary consumers. It has herbivores, predators, scavengers, and decomposers. It has competition, predation, parasitism, and likely mutualism. It is, in miniature, a fully functioning biosphere, one that has been operating independently for 5.5 million years. The traits shared by the animals of Movile Cave tell a story of convergent adaptation of different lineages, facing the same environmental challenges, arriving at the same evolutionary solutions. This phenomenon, known as troglomorphism, is one of the most consistent patterns in cave biology, observed in subterranean ecosystems around the world. But in Movile Cave, these adaptations are particularly pronounced, reflecting the extreme antiquity and severity of the cave's isolation. The most obvious troglomorphic trait is the loss of eyes. In a world of permanent, absolute darkness, eyes are not just useless, they are a liability. Eyes are complex organs that require significant energy to develop and maintain. They are vulnerable to infection and injury. They take up space on the head that could be devoted to other, more useful sensory structures. In the absence of any light to detect, natural selection does not maintain the genes responsible for eye development, and over many generations, these genes accumulate mutations that progressively reduce and eventually eliminate the eyes. This is not a Lamarckian process. It is not that cave animals lost their eyes through disuse. Rather, it is that in the absence of selection pressure to maintain functional eyes, random mutations that degrade eye development are no longer weeded out. They accumulate, they spread through the population. Over thousands of generations, the eyes shrink, become covered by skin, and eventually disappear entirely. Some of the Movile Cave species show intermediate stages of this process: reduced eyes covered by a thin membrane, while others have lost all external trace of visual organs. The loss of pigmentation follows a similar logic. Pigmentation serves multiple functions in surface organisms: protection from ultraviolet radiation, camouflage from predators, signaling to potential mates. In a cave, none of these functions have any relevance. There is no ultraviolet light to protect against. There are no visually hunting predators to hide from. There are no mates to impress with colorful displays. The genes responsible for pigment production are freed from selection pressure, and mutations that reduce pigmentation accumulate unchecked. The result is the ghostly, translucent appearance of cave animals, bodies so pale that internal organs are sometimes visible through the skin. This is not albinism in the clinical sense, though the end result is superficially similar. It is a gradual, evolutionary depigmentation driven by the relaxation of selection for color. While eyes and pigment are being lost, other structures are being enhanced. Many cave animals develop elongated appendages, longer legs, longer antennae, longer sensory hairs. In a world without sight, touch becomes the primary sense, and longer appendages increase the animal's ability to detect objects, surfaces, and other organisms at a distance. The antennae of Movile Cave's crustaceans are notably longer than those of their surface relatives, providing an enhanced tactile awareness that compensates for the absence of vision. Chemoreception, the ability to detect chemical signals, is also enhanced in many cave organisms. In the dark, the ability to smell or taste the environment becomes critically important for finding food, avoiding predators, locating mates, and navigating through complex passage networks. Many cave animals have more chemoreceptive sensory structures than their surface counterparts, and behavioral studies suggest that they are more sensitive to chemical cues. Vibration sensitivity is another enhanced trait. In the still, quiet environment of a cave, vibrations transmitted through rock, water, or air can carry information about the presence and movement of other organisms. Many cave animals have specialized sensory organs, mechanoreceptors, that detect minute vibrations, giving them a sort of seismic awareness of their surroundings. These adaptations, the loss of unnecessary structures and the enhancement of useful ones, represent an economization of energy. In the energy-limited environment of Movile Cave, every calorie counts. Organisms that waste energy on maintaining useless eyes or producing unnecessary pigment are at a disadvantage compared to those that redirect that energy toward more productive purposes: better sensory organs, more efficient metabolism, greater reproductive output. Over millions of years, natural selection has ruthlessly pruned the unnecessary and amplified the essential, sculpting organisms that are exquisitely adapted to their specific environment and, correspondingly, hopelessly unfit for any other. This is one of the great paradoxes of cave adaptation. The very traits that make cave organisms so successful underground make them utterly unable to survive on the surface. They cannot tolerate bright light. They cannot compete with pigmented, sighted organisms. They cannot regulate their body temperature in a fluctuating surface environment. They are specialists of the highest order, perfectly adapted to a single, narrow niche and completely unable to function outside of it. The evolutionary history of Movile Cave's inhabitants is a subject of intense scientific interest and ongoing research. How did these organisms get into the cave? When did they arrive? How have they changed since their isolation? These questions require detective work of the highest order, combining morphological analysis, molecular phylogenetics, biogeography, and geological reconstruction. The prevailing hypothesis is that the ancestors of Movile Cave's current inhabitants were surface-dwelling or soil-dwelling organisms that entered the cave system before it was sealed, approximately 5.5 million years ago, during the dramatic environmental upheavals of the Messinian salinity crisis. During this period, the desiccation of the Mediterranean Sea caused profound changes in climate, hydrology, and habitat across southeastern Europe. Many surface habitats were disrupted or destroyed, and organisms that had previously lived in soil, springs, and shallow groundwater may have been driven underground, following retreating water tables into deeper and more stable subterranean environments. Some of these organisms found their way into the cave system that would become Movile Cave. When the cave was subsequently sealed by accumulating clay and less deposits, these organisms were trapped inside, cut off from the surface permanently, forced to adapt to their new, lightless, chemically extreme environment or perish. Molecular phylogenetic studies, analyses that use DNA sequences to reconstruct evolutionary relationships and estimate divergence times, have generally supported this timeline. Comparisons between Movile Cave species and their closest surface-dwelling relatives suggest divergence times consistent with several million years of isolation. The cave's amphipods, for example, appear to have diverged from their nearest surface relatives approximately 5 to 6 million years ago, consistent with the geological estimate of the cave's sealing. However, not all of the cave's inhabitants necessarily arrived at the same time. Some species may have entered earlier, during initial stages of cave formation, while others may have arrived later, just before the final sealing. Some may have entered through the deep thermal water system rather than from the surface, ascending from deeper underground environments where they had already been living a subterranean existence. This possibility is particularly relevant for the cave's aquatic organisms, which may have ancestral connections to deep groundwater faunas rather than surface faunas. The question of whether any organisms have entered or left the cave since its sealing is also important. The geological and geochemical evidence strongly suggests that the seal has been essentially complete, no significant exchange of air, water, or organisms with the surface. But essentially complete is not the same as absolutely perfect. It is possible that over 5.5 million years, occasional minor breaches in the seal may have allowed limited exchanges, perhaps a few microorganisms entering with slowly percolating groundwater, or a few gases diffusing through the clay cap. But these would have been minuscule transient events, insufficient to significantly alter the cave's biological or chemical isolation. Inside the cave, once isolated, the organisms underwent a remarkable evolutionary radiation. From a relatively small number of ancestral colonizers, the cave's fauna diversified into the 50-plus species present today. This diversification was driven by the same processes that drive speciation everywhere: genetic drift, natural selection, adaptation to different microhabitats, and reproductive isolation between populations. The cave, despite its small size, offers several distinct microhabitats that organisms can specialize in. The air-water interface, where microbial mats are thickest and food is most abundant. The open water column, where planktonic organisms drift and swim. The submerged cave floor with its sediment and debris. The dry cave walls and ceiling, where terrestrial organisms climb and hunt. The narrow crevices and fissures, accessible only to the smallest and most flexible organisms. Each of these microhabitats presents different challenges and opportunities, and organisms that specialize in one may become reproductively isolated from those that specialize in another, eventually diverging into separate species. The result is a surprisingly high diversity of species for such a small space, a diversity born not of geographic vastness, as in the Galapagos Islands, but of microhabitat partitioning within a confined environment. Movile Cave is a kind of evolutionary laboratory where the usual drivers of speciation, geographic isolation, ecological differentiation, sexual selection have operated within a space measured in meters rather than kilometers. The concept of evolutionary filtering is central to understanding the Movile Cave ecosystem. The cave's environment is not just dark, it is chemically extreme. The high levels of hydrogen sulfide, the low oxygen, the elevated carbon dioxide, and the warm, humid conditions create a filter that excludes most organisms and admits only those capable of tolerating these conditions. This filter operates at every level, from the molecular to the organismal. At the molecular level, hydrogen sulfide is toxic because it inhibits cytochrome C oxidase, a critical enzyme in the mitochondrial electron transport chain, which is responsible for aerobic respiration. When cytochrome C oxidase is inhibited, cells cannot use oxygen to produce energy, and they die. This is the same mechanism by which hydrogen sulfide is lethal to humans and most other aerobic organisms. The organisms of Movile Cave have evolved various strategies to cope with this toxicity. Some have modified versions of cytochrome C oxidase that are less sensitive to sulfide inhibition. Others have developed biochemical pathways that detoxify sulfide before it can reach the mitochondria. For example, enzymes that oxidize sulfide to less toxic sulfur compounds like thiosulfate or elemental sulfur. Still others maintain elevated levels of antioxidants and sulfide-binding proteins that buffer the intracellular concentration of free sulfide. These adaptations are not trivial. They represent fundamental changes in cellular biochemistry, changes that have been refined over millions of years of exposure to chronic sulfide stress. They are analogous to the adaptations seen in organisms living near deep-sea hydrothermal vents, where hydrogen sulfide is also abundant. In both cases, life has found ways to cope with a chemical that is fundamentally incompatible with the normal biochemistry of aerobic organisms. At the organismal level, the evolutionary filter has selected for organisms with low metabolic rates, organisms that require less oxygen and produce less metabolic waste. In the oxygen-poor atmosphere of Movile Cave, organisms that can function on minimal oxygen have a significant advantage. Many of the cave's animals appear to have metabolic rates substantially lower than their surface relatives. They move more slowly, grow more slowly, and reproduce more slowly, but they survive in conditions that would quickly kill a more metabolically active organism. The filter has also selected for organisms with efficient nutrient recycling capabilities. In an energy-limited environment, every atom of carbon, nitrogen, phosphorus, and other essential elements is precious. Organisms that can efficiently extract nutrients from their food, minimize waste, and recycle organic matter have an advantage over those that are more profligate. The tight nutrient cycling within the Movile Cave ecosystem, with decomposers rapidly breaking down dead organisms and returning their nutrients to the microbial base of the food web, is a reflection of this selective pressure. The evolutionary filter also operates through competitive exclusion. In the confined environment of the cave, resources are limited and competition is intense. Organisms that cannot compete effectively for food, space, or mates are eliminated. Over millions of years, this competition has winnowed the fauna down to a relatively small number of highly specialized species, each occupying a distinct ecological niche and each supremely adapted to its particular role in the ecosystem. The result of this filtering is an ecosystem that is simplified compared to most surface ecosystems: fewer species, fewer trophic levels, less redundancy, but one that is remarkably stable and efficient. Every organism has a role, every niche is filled, every calorie is accounted for. It is a lean, elegant system, stripped of the biological excess that characterizes more resource-rich environments, and refined by millions of years of selection into a state of near-perfect adaptation to its unique conditions. The relationship between the cave's animals and its chemosynthetic bacteria deserves special attention because it is not simply a matter of animals eating bacteria. In many cases, the relationship is more intimate and more complex, a symbiosis in which bacteria live within or upon the bodies of animal hosts, providing metabolic services in exchange for shelter, nutrients, or other benefits. The most well-studied example of symbiosis in chemosynthetic ecosystems comes from deep-sea hydrothermal vents, where giant tube worms of the genus Riftia harbor dense populations of sulfur-oxidizing bacteria within a specialized organ called the trophosome. These bacteria provide the tube worms with all of their nutrition. The worms have no mouth, no gut, and no anus. They are entirely dependent on their bacterial symbionts for food. Movile Cave, similar, though less extreme, symbiotic relationships appear to exist. Several of the cave's invertebrate species have been found to harbor sulfur-oxidizing bacteria on their body surfaces or within their tissues. These bacteria may serve multiple functions. They may provide the host with supplementary nutrition by synthesizing organic compounds from hydrogen sulfide. They may help detoxify the host's immediate environment by converting toxic sulfide to less harmful forms. They may provide the host with essential vitamins or amino acids that are difficult to obtain from the limited food sources available in the cave. The details of these symbiotic relationships are still being elucidated. The difficulty of studying organisms in such a restricted and hostile environment means that many questions remain unanswered. How are the symbiotic bacteria transmitted from parent to offspring? Are they acquired from the environment each generation, or are they inherited? How tightly co-adapted are the partners? Could the host survive without its symbionts, or are the two now inseparable? These are questions that researchers are actively investigating, using a combination of microscopy, molecular biology, and experimental approaches. What is clear, however, is that symbiosis plays an important role in the Movile Cave ecosystem, just as it does in deep-sea vent ecosystems, in coral reefs, in tropical forests, and in virtually every other ecosystem on Earth. Life, it seems, is inherently collaborative. Even in the most extreme environments, organisms find ways to form partnerships that benefit all parties, creating integrated units of survival that are more effective than any single species could be alone. The parallels between Movile Cave and deep-sea hydrothermal vents are striking and instructive. Both are ecosystems powered by chemosynthesis rather than photosynthesis. Both are based on the oxidation of hydrogen sulfide and other reduced chemicals from geological sources. Both harbor communities of organisms adapted to high sulfide, low oxygen, and elevated temperatures. Both feature symbiotic relationships between animals and chemosynthetic bacteria. And both were completely unknown to science until relatively recently. Deep-sea vents were discovered in 1977, just 9 years before Movile Cave. These parallels suggest that chemosynthetic ecosystems may be far more common on Earth than previously recognized. For most of the history of biology, scientists assumed that all complex ecosystems required sunlight. The discovery of hydrothermal vent communities in the late 1970s shattered that assumption. The discovery of Movile Cave's ecosystem in the late 1980s reinforced the new paradigm. And since then, additional chemosynthetic communities have been found in other caves, in deep continental subsurface environments, in marine sediments, and in subglacial lakes beneath polar ice sheets. The emerging picture is one of a planet with two biospheres, a surface biosphere powered by the sun, and a subsurface biosphere powered by the Earth's internal chemistry. The surface biosphere is the one we are familiar with, the one we can see, touch, and photograph. It is diverse, productive, and visually spectacular. But the subsurface biosphere may be equally vast, equally ancient, and equally important to the overall functioning of Earth's biological systems. And Movile Cave offers one of the clearest and most accessible windows into this hidden world. Let us now consider the specific mechanisms by which energy flows through the Movile Cave ecosystem, the food web that connects the chemosynthetic bacteria at the base to the predators at the top. The food web begins with the chemoautotrophic bacteria, the primary producers. These organisms fix carbon dioxide into organic compounds using energy derived from the oxidation of hydrogen sulfide, methane, and ammonium. The organic compounds they produce, sugars, amino acids, lipids, and other molecules, represent the total energy input to the ecosystem. This is the cave's gross primary production, and everything else depends on it. The primary producers are consumed by primary consumers, organisms that feed directly on bacterial mats or on individual bacterial cells. In the aquatic zone, the primary consumers include the amphipods, isopods, and copepods that graze on the microbial mats floating at the water's surface or coating submerged surfaces. These small crustaceans scrape bacteria from surfaces with specialized mouthparts, ingesting the organic-rich microbial material and extracting the energy stored in its chemical bonds. In the terrestrial zone, primary consumers include springtails and mites that feed on bacterial and fungal films growing on the cave walls and floor. These tiny arthropods are the terrestrial equivalents of the aquatic grazers, converting microbial biomass into animal biomass and making it available to higher trophic levels. The primary consumers are in turn eaten by secondary consumers, predators that feed on the grazers. In the aquatic zone, the blind leech Hemopes kika is a key secondary consumer, preying on amphipods and other small crustaceans. The water scorpion Nepa anophthalma is another aquatic predator, using its raptorial forelegs to seize and subdue prey. In the terrestrial zone, the spiders and pseudoscorpions are the primary predators, catching springtails, mites, and other small arthropods. These predators represent the top of the food chain in their respective zones. There is no tertiary predator above them, no apex carnivore that feeds on the spider, that feeds on the springtail, that feeds on the bacteria. The food chain is short, typically consisting of only 3 trophic levels: producers, primary consumers, and secondary consumers. This short food chain is a direct consequence of the limited energy available from chemosynthesis. At each trophic level, energy is lost to metabolism. The organisms use most of the energy they consume to fuel their own life processes, with only a small fraction being converted into new biomass that is available to the next trophic level. The typical ecological efficiency, the percentage of energy passed from one trophic level to the next, is roughly 10%. This means that if the primary producers fix 100 units of energy, only about 10 units are available as primary consumer biomass, and only about 1 unit is available as secondary consumer biomass. In an energy-rich surface ecosystem, this cascading energy loss can support many trophic levels, 4, 5, or even 6 in some marine food webs. But in the energy-poor environment of Movile Cave, the total primary production is so low that only 2 or 3 trophic levels can be sustained. There simply is not enough energy to support a 4th level. This is why there are no tertiary predators in the cave. The energy has been used up by the time it reaches the top of the short food chain. The food web also includes decomposers, organisms that feed on dead organic matter, breaking down the remains of bacteria, animals, and other organisms, and returning their constituent nutrients to the ecosystem. In Movile Cave, fungi and certain bacteria serve as the primary decomposers, breaking down dead biomass through enzymatic digestion and absorbing the resulting nutrients. This decomposition is critically important because it recycles nutrients that would otherwise be permanently locked up in dead organic matter, unavailable to living organisms. The tight coupling between production and decomposition is one of the defining features of the Movile Cave ecosystem. Nothing is wasted. Dead organisms are rapidly broken down. Nutrients are quickly recycled. The same atoms of carbon, nitrogen, and phosphorus cycle through the ecosystem again and again, passing from bacteria to grazers to predators to decomposers and back to bacteria. This efficient recycling is what allows a complex ecosystem to persist on such a limited energy budget. Every nutrient is used and reused, maximizing the biological productivity that can be extracted from the cave's modest chemical energy supply. One of the most intriguing aspects of the Movile Cave ecosystem is the role of the cave's atmosphere, specifically the thin layer of air trapped between the water's surface and the cave ceiling. This air layer is not just a space that terrestrial organisms inhabit, it is an active participant in the cave's chemistry and biology. The composition of this trapped air, as we have discussed, is dramatically different from the surface atmosphere, but it is not static. The air composition fluctuates over time, driven by the ongoing chemical and biological processes within the cave. Microbial activity consumes oxygen and produces carbon dioxide. Chemical reactions between hydrogen sulfide and oxygen remove both gases and produce sulfuric acid, which dissolves the cave's limestone walls, slowly enlarging the passages over geological time. Methane enters the atmosphere from the thermal waters and is consumed by methanotrophic bacteria in the microbial mats and on the cave walls. These processes create a dynamic chemical environment, a constantly shifting balance of gases that the cave's organisms must navigate. The oxygen concentration, for instance, is not uniform throughout the cave. It tends to be slightly higher near the artificial access shaft, where small amounts of surface air can diffuse in, and lower in the more remote chambers, where biological oxygen consumption has had longer to deplete the supply. These oxygen gradients create microzones within the cave, areas of slightly higher oxygen where organisms with higher metabolic demands can thrive, and areas of lower oxygen where only the most tolerant species can survive. The hydrogen sulfide concentration also varies spatially and temporally. It is highest near the thermal water sources, where fresh sulfide is constantly being introduced, and lower in areas farther from these sources, where oxidation has had time to reduce the sulfide concentration. Animals that are sensitive to sulfide tend to avoid the most sulfide-rich areas, while those with effective sulfide detoxification mechanisms can exploit these high sulfide zones where competition is reduced. This spatial heterogeneity in atmospheric composition is another factor contributing to the cave's biodiversity. By creating multiple microenvironments with different chemical conditions, the cave provides niches for organisms with different tolerances and capabilities. A species that can tolerate high sulfide but requires more oxygen occupies a different niche than one that can tolerate low oxygen but is sensitive to sulfide. This niche differentiation reduces competition and allows more species to coexist, a principle known as the competitive exclusion principle, which states that two species competing for exactly the same resources cannot coexist indefinitely. In Movile Cave, the chemical heterogeneity of the environment ensures that different species face different conditions, reducing direct competition and promoting diversity. The water chemistry of Movile Cave is equally complex and equally important to the ecosystem's functioning. The thermal waters that rise into the cave from deep underground are not pure water. They are complex chemical solutions carrying dissolved gases, minerals, and ions that reflect their long journey through the Earth's crust. The temperature of these waters is around 21 degrees Celsius, warm for groundwater in this region, which typically averages 10 to 12 degrees. This warmth is of geothermal origin, derived from the Earth's internal heat, which increases with depth at a rate of approximately 25 to 30 degrees per kilometer in this geological setting. The thermal waters in Movile Cave likely originate from depths of several hundred meters or more, where they have been heated by the surrounding rock and have had time to dissolve various minerals and gases. The most important dissolved gases are hydrogen sulfide, methane, and carbon dioxide, the chemical substrates and products of the cave's chemosynthetic processes. These gases are present in concentrations far above what would be found in surface waters, reflecting the deep geological origin of the thermal waters and the absence of atmospheric ventilation that would normally strip dissolved gases from surface water bodies. The water is also rich in dissolved minerals, calcium, magnesium, sulfate, chloride, bicarbonate, and various trace elements. The exact mineral composition depends on the geology of the aquifer from which the water originates and the chemical reactions that occur as the water travels through different rock layers. These dissolved minerals serve as nutrients for the cave's organisms and influence the cave's geological processes. For example, the precipitation of calcium carbonate, forming stalactites and other speleothems, or the dissolution of limestone by sulfuric acid produced from hydrogen sulfide oxidation. The pH of the cave's water is variable, reflecting the complex interplay of acid-producing and acid-consuming reactions. In areas where hydrogen sulfide is being actively oxidized to sulfuric acid, the pH can be quite low, acidic enough to dissolve limestone. In other areas, the buffering capacity of the dissolved carbonates maintains a more neutral pH. These pH variations create yet another dimension of chemical heterogeneity within the cave, further diversifying the available niches and supporting a wider range of microbial and animal communities. One particularly fascinating aspect of the water chemistry is the sulfur cycle, the set of chemical and biological transformations that sulfur undergoes as it passes through the cave ecosystem. Sulfur enters the cave as hydrogen sulfide dissolved in the thermal waters. Chemosynthetic bacteria oxidize this sulfide, producing elemental sulfur, sulfite, thiosulfate, and ultimately sulfate. The sulfate may be reduced back to sulfide by anaerobic sulfate-reducing bacteria in the deeper, oxygen-free zones of the microbial mats and sediments. The reduced sulfide is then available for ray oxidation, completing the cycle. This sulfur cycle is analogous to the carbon, nitrogen, and phosphorus cycles that operate in surface ecosystems, but it has a distinctly subterranean character. The sulfur is not being cycled between the atmosphere, the oceans, and the biosphere as it is on the surface. It is being cycled within a closed system, between the thermal waters, the microbial communities, and the cave's geological matrix. This tight internal cycling is characteristic of the cave's overall nutrient dynamics, a closed loop, with minimal inputs and minimal losses, sustained by the steady supply of chemical energy from the deep earth. The question of how the Movile Cave ecosystem sustains itself over geological time, millions of years, raises important issues about long-term stability, resilience, and the minimum conditions required for ecosystem persistence. Surface ecosystems are subject to constant disturbance: storms, droughts, fires, volcanic eruptions, ice ages, asteroid impacts. These disturbances can dramatically alter the structure and composition of biological communities, driving some species to extinction and opening opportunities for others. The history of life on Earth's surface is a history of boom and bust, of catastrophic die-offs and explosive radiations, of constant change driven by an unpredictable environment. Movile Cave, by contrast, has been buffered from virtually all of these external disturbances. The cave's temperature, humidity, and water chemistry have likely remained relatively stable for millions of years, modulated by the slow, steady flow of geothermal waters rather than by the rapid fluctuations of weather and climate. There have been no storms, no droughts, no fires, no ice ages. The Pleistocene glaciations that reshaped the surface of Europe and drove countless species to extinction or migration passed over Movile Cave without any detectable effect. The cave's organisms were insulated from the climatic upheavals that occurred above them, living in a thermal and chemical cocoon that changed only on geological timescales. This stability has profound implications for the cave's evolutionary dynamics. In a stable environment, the rate of evolutionary change tends to slow. There are fewer novel challenges to adapt to, fewer new niches to exploit, fewer selection pressures to drive the evolution of new traits. The organisms of Movile Cave may have reached a state of near evolutionary equilibrium, a condition in which the rate of adaptive evolution has slowed to a minimum because the organisms are already well adapted to their environment and the environment is not changing. This does not mean that evolution has stopped in Movile Cave. Genetic drift continues to operate, causing random changes in gene frequencies that accumulate over time. Sexual selection may drive changes in mate preferences and reproductive behavior. Parasites and pathogens impose ongoing selection pressures on their hosts, but the broad outlines of the ecosystem, the major species, the trophic structure, the metabolic strategies, may have been relatively unchanged for a very long time, perhaps millions of years. This long-term stability makes Movile Cave a kind of living fossil, not in the sense that its organisms are primitive or unchanged from their ancestors, but in the sense that the ecosystem as a whole may have persisted in approximately its current form for an extraordinarily long time. It is a snapshot of a biological community that reached a stable configuration millions of years ago and has been ticking along ever since, powered by the same chemical energy sources, following the same ecological rules, running on the same slow biological clock. The study of Movile Cave has been challenging from the very beginning, and the logistical difficulties of working in the cave have shaped and limited our understanding of its ecosystem in important ways. As mentioned earlier, access to the cave is severely restricted. The Romanian government and the scientific community have agreed that the cave's protection must take priority over research, and entry is limited to a small number of carefully vetted scientists per year. Each visit is brief, typically a few hours at most, and is conducted under strict protocols designed to minimize human impact. Researchers wear sterilized clothing, carry self-contained breathing apparatus, and avoid touching cave surfaces whenever possible. The hostile atmospheric conditions further limit what researchers can accomplish during their brief visits. The low oxygen, high carbon dioxide, and hydrogen sulfide exposure cause physiological symptoms that impair cognitive and physical function. Taking samples, making measurements, and recording observations must all be done quickly and efficiently, with minimal equipment and under significant physical and mental stress. Laboratory analysis of samples brought out of the cave presents its own challenges. Many of the cave's organisms are adapted to the specific chemical conditions inside the cave: the low oxygen, the hydrogen sulfide, the warm temperature, and they do not survive well under standard laboratory conditions. Culturing bacteria from the cave has proven difficult, as many species require specific, hard-to-replicate chemical environments to grow. Keeping animal specimens alive outside the cave is equally challenging, and most observations of animal behavior and ecology must be made in situ, during the limited time available on each visit. Despite these challenges, decades of research have produced a substantial body of knowledge about the cave and its inhabitants. Romanian scientists, led initially by Cristian Lascu and later by a growing international community of collaborators, have catalogued the cave's species, characterized its atmospheric and water chemistry, analyzed the isotopic composition of its organic matter, and conducted molecular phylogenetic studies of its key organisms. Isotopic analysis has been particularly informative. By measuring the ratios of stable carbon isotopes, carbon-13 to carbon-12, in the cave's organic matter, researchers have been able to trace the flow of carbon through the food web and confirm that the ecosystem is indeed powered by chemosynthesis rather than by organic matter imported from the surface. Organic matter produced by photosynthesis has a distinctive carbon isotope signature that differs from that produced by chemosynthesis. The organic matter in Movile Cave has a chemosynthetic signature throughout, from the microbial mats to the tissue of the cave's predators, confirming that the entire food web is built on chemosynthetically fixed carbon. Nitrogen isotope analysis has provided complementary information about trophic structure, the feeding relationships within the food web. As nitrogen passes up the food chain from bacteria to grazers to predators, the ratio of nitrogen-15 to nitrogen-14 increases in a predictable way, with each trophic level showing a characteristic enrichment in nitrogen-15. By measuring the nitrogen isotope ratios in different organisms, researchers have been able to map out the trophic structure of the cave's food web, confirming the roles of different species as producers, primary consumers, or secondary consumers. Molecular studies using DNA sequencing have revealed the phylogenetic relationships of the cave's organisms, their degree of divergence from surface relatives, and the diversity of the microbial communities. Metagenomics, the study of DNA extracted directly from environmental samples, without the need to culture individual organisms, has been particularly powerful, revealing a microbial diversity far greater than what could be detected by traditional culture-based methods. These studies have revealed that the microbial community in Movile Cave includes hundreds of distinct species, many of them previously unknown to science. The metabolic capabilities of these microorganisms are diverse, encompassing sulfur oxidation, methane oxidation, nitrification, sulfate reduction, fermentation, and numerous other biochemical pathways. This metabolic diversity is the engine that drives the cave's ecosystem, a complex, interlocking network of chemical reactions that converts the cave's geological energy supply into biological energy. The thermal waters that feed Movile Cave deserve further exploration, because they are the ultimate source of the cave's energy and the key to its long-term sustainability. These waters do not originate within the cave itself. They rise from deep underground, driven by geological forces that have been operating for far longer than the cave has been sealed. The geological setting of the Dobrogea region includes deep fault systems, fractures in the Earth's crust that extend to considerable depths. These faults act as conduits, allowing water to circulate between the surface and the deep subsurface. Rainwater that infiltrates the ground far from the cave may travel downward along these fault systems, reaching depths of several hundred meters to a few kilometers, where it is heated by the Earth's geothermal gradient and enriched in dissolved gases and minerals by contact with deep rock formations. At depth, the water encounters geological formations rich in sulfide minerals, pyrite, marcasite, and other iron-sulfur compounds. Chemical reactions between the water and these minerals release hydrogen sulfide into solution. The water also encounters formations rich in organic matter, ancient sediments containing the remains of long-dead organisms, which produce methane through slow, thermally driven decomposition. And the water dissolves ammonium from nitrogen-rich minerals and organic deposits. This enriched water then rises back toward the surface, driven by hydrostatic pressure, buoyancy. Heated water is less dense than cold water, and geological forces. It enters the Movile Cave system through fractures in the cave floor, bringing with it the chemical substrates, hydrogen sulfide, methane, ammonium, that fuel the cave's chemosynthetic ecosystem. This process has been going on for millions of years, and there is no reason to think it will stop anytime soon. The geological forces that drive the circulation of thermal waters operate on timescales of tens of millions to hundreds of millions of years. The sulfide minerals in the deep rock formations are vast reservoirs of chemical energy that will not be exhausted for geological ages. The geothermal gradient that heats the water is a consequence of the Earth's internal heat generated by the radioactive decay of uranium, thorium, and potassium in the Earth's mantle and crust, a process that will continue for billions of years. In other words, the energy supply of Movile Cave is effectively inexhaustible on biological timescales. The cave could potentially sustain its ecosystem for hundreds of millions of years, far longer than the 5.5 million years it has already been sealed. This is a remarkable thought. It means that if left undisturbed, the organisms of Movile Cave could continue evolving in isolation for time periods comparable to the entire history of complex life on Earth. What would they become? How much further could evolution push them? These are questions that we cannot answer, but they inspire a profound sense of wonder about the potential of life to persist and diversify in the most unlikely of places. The role of sulfuric acid in shaping the cave itself is worth pausing to consider, because it connects the biological and geological aspects of the Movile Cave system in a way that is both elegant and revealing. When chemosynthetic bacteria oxidize hydrogen sulfide, one of the end products is sulfuric acid, a strong mineral acid that is highly corrosive to limestone. This sulfuric acid dissolves the calcium carbonate that makes up the cave's walls, ceiling, and floor, converting it to calcium sulfate, gypsum, and carbon dioxide. This process is known as sulfuric acid speleogenesis, and it is responsible for the formation and enlargement of some of the world's most spectacular caves, including Leeches Cave and Carlsbad Caverns in the western regions of North America. In these caves, hydrogen sulfide rising from deep petroleum deposits was oxidized by oxygen-containing groundwater, producing sulfuric acid that dissolved enormous volumes of limestone and created vast underground chambers. In Movile Cave, the same process is occurring, but on a much smaller scale. The cave is not a vast underground cathedral. It is a modest system of narrow passages and small chambers. But the sulfuric acid produced by the cave's chemosynthetic bacteria is actively dissolving limestone, slowly enlarging the cave and modifying its shape over time. This creates a fascinating feedback loop between biology and geology. The bacteria produce sulfuric acid as a byproduct of their energy-generating metabolism. The sulfuric acid dissolves limestone, creating more space for the bacteria to colonize. The increased surface area supports more bacteria, which produce more sulfuric acid, which dissolves more limestone. Biology is literally reshaping the physical environment, and the reshaped environment, in turn, supports more biology. This kind of feedback, where organisms modify their physical environment in ways that benefit themselves, is known as ecosystem engineering, and it is observed in many environments, from beaver dams to coral reefs to termite mounds. In Movile Cave, the ecosystem engineers are microscopic bacteria, and their construction project is a cave, a slow, patient dissolution of rock that has been proceeding millimeter by millimeter for millions of years. The gypsum produced by the reaction between sulfuric acid and limestone accumulates on the cave walls in characteristic formations, crusts, crystals, and deposits that provide a geological record of the ongoing chemical activity. By analyzing these gypsum deposits, their thickness, their isotopic composition, their crystal structure, researchers can reconstruct the history of sulfuric acid production in the cave, and by extension, the history of chemosynthetic activity. This geological record suggests that chemosynthesis has been occurring in the Movile Cave system for a very long time, at least as long as the cave has been sealed, and possibly longer. The bacteria were not newcomers when the cave was isolated. They were already there, already oxidizing sulfide, already producing sulfuric acid, already dissolving limestone. The sealing of the cave did not create the chemosynthetic ecosystem. It merely isolated it, cutting it off from the surface and allowing it to evolve independently for millions of years. Let us now return to the animals of Movile Cave and examine some of the specific species in greater detail, because it is in the details of their anatomy, physiology, and behavior that the full power of evolution in isolation becomes most apparent. Consider the blind leech, Hemopes kika. This animal belongs to the family Hemippidae, a group of leeches found in freshwater habitats throughout Europe and Western Asia. Surface-dwelling Hemopes species are predators, feeding on earthworms, snails, insect larvae, and other invertebrates. They have well-developed eyes, typically 5 pairs of simple eyes arranged on the head, which they use to detect light and shadow, helping them locate prey and navigate their environment. Hemopes kika has no eyes. Not reduced eyes, not vestigial eyes, no eyes at all. The position on the head where eyes would normally be located is smooth skin, with no trace of visual organs. This complete loss of eyes is consistent with millions of years of evolution in total darkness, enough time for the genes responsible for eye development to accumulate mutations and become non-functional. But the blind leech has compensated for the loss of vision with enhanced mechanorception and chemoreception. Its body surface is densely covered with sensory papillae, small, raised bumps containing clusters of sensory neurons that detect touch, water currents, vibrations, and chemical signals. These papillae are more numerous and more sensitive than those of surface Hemopes species, providing the blind leech with a detailed, non-visual perception of its surroundings. Behavioral studies have shown that the blind leech is an effective predator despite its lack of vision. It detects prey primarily through vibrations in the water. The movements of a swimming amphipod or isopod create pressure waves that the leech can detect and home in on. Chemical cues, dissolved amino acids, and other organic molecules released by potential prey also play a role, allowing the leech to detect the presence of food at a distance and orient toward it. The blind leech's body is elongated and highly flexible, allowing it to navigate the narrow, irregular passages of the flooded cave chambers. Its body is unpigmented, a uniform pale color, almost translucent, and its skin is thin, allowing for cutaneous respiration, the absorption of oxygen directly through the skin's surface. This is an important adaptation in the cave's low oxygen environment, where every available mechanism for oxygen uptake is needed. The feeding behavior of Hemopes kika is characterized by a slow, methodical approach. The leech does not chase its prey. In the low-energy environment of the cave, sustained pursuit would be metabolically costly and inefficient. Instead, it waits, sensing the water for vibrations and chemical signals, and then moves slowly toward the detected prey, striking when it is close enough to ensure a successful capture. This ambush strategy minimizes energy expenditure while maximizing the probability of a meal, a strategy well suited to an environment where calories are scarce and every feeding opportunity matters. Now consider the water scorpion, Nepa anophthalma. This insect belongs to the family Nepidae, a group of aquatic predatory bugs found in freshwater habitats worldwide. Surface Nepa species are ambush predators, lurking among submerged vegetation and seizing passing prey with their powerful, mantis-like forelegs. They breathe through a long, siphon-like tube extending from the rear of the abdomen to the water surface. Nepa anophthalma retains this basic body plan but has been modified in characteristic ways by its long sojourn in the cave. Like the blind leech, it has completely lost its eyes. Its body is unpigmented. Its forelegs retain their raptorial function. They are still powerful grasping appendages used to capture prey, but they may be proportionally longer than in surface species, increasing the animal's reach and allowing it to capture prey from a greater distance in the dark. The breathing siphon of Nepa anophthalma presents an interesting adaptive challenge. On the surface, water scorpions use their siphon to breathe normal air, 21% oxygen, minimal toxic gases. In Movile Cave, the air above the water is depleted in oxygen and enriched in hydrogen sulfide and carbon dioxide. Breathing through a siphon in this atmosphere means inhaling gases that are both insufficient in oxygen and actively toxic. The water scorpion must have physiological mechanisms to cope with these challenges, perhaps a more efficient oxygen transport system, or sulfide detoxifying enzymes in its tissues, or behavioral strategies to minimize exposure to the most toxic air pockets. These are questions that remain largely unanswered because the extreme difficulty of studying live animals in the cave limits the type of physiological investigations that can be conducted. But the very existence of Nepa anophthalma, a fully functioning predatory insect living in an atmosphere that would kill most insects in minutes, testifies to the extraordinary power of natural selection to adapt organisms to even the most hostile environments, given enough time. The spiders of Movile Cave are equally remarkable. Several species have been identified, including members of the genus Agricina, small hunting spiders that do not build webs but instead actively search for prey on the cave walls and floor. These spiders are eyeless and pale, with elongated legs that give them a spidery, ethereal appearance even by spider standards. In the total darkness of the cave, these spiders cannot use vision to detect prey. Instead, they rely on vibrations transmitted through the substrate, the cave walls and floor, and through the air. Their leg joints contain slit sensilla, specialized sensory organs that detect minute vibrations, and their bodies are covered with trichobothria, fine hair-like structures that detect air currents and the sound waves produced by the movements of potential prey. A springtail jumping on the cave floor creates a pattern of vibrations that radiates outward through the rock. A spider several centimeters away detects these vibrations through its slit sensilla, orients toward the source, and approaches cautiously, guided by the vibrational signals. When close enough, the spider lunges, seizing the prey with its chelicerae, its fangs, and injecting venom to subdue it. This hunting strategy requires a level of sensory sophistication that is truly impressive. The spider must be able to distinguish between the vibrations produced by prey, which are worth pursuing, and the vibrations produced by other objects, falling water drops, settling rock, the movements of other predators, which are not. It must be able to determine the distance and direction of the vibration source, and it must be able to calibrate its approach to maximize the probability of capture while minimizing energy expenditure. All of this must be done without any visual information whatsoever, purely through the interpretation of mechanical signals. The evolution of such refined sensory capabilities is a testament to the power of natural selection in an extreme environment. In Movile Cave, every calorie counts. A spider that wastes energy pursuing non-prey vibrations, or that fails to detect a potential meal passing nearby, is at a significant disadvantage. Over millions of years, selection has honed the sensory capabilities of these spiders to a remarkable degree, producing animals that are, in their own domain, as effective as any sighted predator. The reproductive biology of Movile Cave's organisms is another area of great interest, though one where our knowledge is particularly limited due to the difficulty of observation. How do organisms that live in total darkness find mates? How do they reproduce? How many offspring do they produce, and how often? For most cave organisms, mate finding relies on chemical signals, pheromones released into the air or water that attract potential mates over distance. In the humid, still atmosphere of Movile Cave, airborne pheromones can persist for extended periods and diffuse over considerable distances, making chemical signaling an effective means of communication. Similarly, in the cave's quiet waters, dissolved pheromones can travel far and maintain their signal strength for a long time. Vibrational signaling may also play a role in mate finding for some species. In surface spiders, for example, males often perform elaborate vibrational courtship displays on the female's web, producing specific patterns of vibrations that identify them as potential mates rather than prey. Similar vibrational courtship behaviors may occur in the cave spider species, though this has not been directly observed due to the logistical constraints of research in the cave. The reproductive rate of cave organisms is generally low compared to their surface relatives. This is a consequence of the energy-limited environment. Producing eggs or offspring requires a significant investment of energy, and in a system where energy is scarce, organisms cannot afford to reproduce as frequently or as prolifically as surface organisms. Cave organisms tend to produce fewer offspring per reproductive event, but invest more energy in each offspring, a strategy known as K-selection, which is typical of organisms living in stable, resource-limited environments. The lifespans of cave organisms may also be extended relative to surface relatives. Several studies of cave-adapted organisms in other cave systems have found that cave species live significantly longer than comparable surface species. This makes sense from an energetic perspective. In an environment where food is scarce and predation pressure is reduced due to the small number of predator species, it is advantageous to live a long time, reproduce slowly, and maximize the total reproductive output over a lifetime rather than reproducing quickly and dying young. In Movile Cave, the specific lifespans and reproductive rates of most species are unknown. But the general pattern, long lives, slow reproduction, low population density, is consistent with the energy constraints of the cave environment and with the K-selected life history strategies observed in other subterranean ecosystems. The genetic structure of Movile Cave's populations is shaped by several factors that are unusual compared to surface populations. The most important of these is the small population size of most species. In a cave system measured in hundreds of meters, there is simply not much space for organisms to live, and the limited energy budget constrains the total number of individuals that can be supported. Small populations are subject to strong genetic drift, random changes in gene frequencies that can, over time, lead to the loss of genetic variation and the fixation of alleles that may not be adaptive. In extreme cases, small populations can experience inbreeding depression, a decline in fitness caused by the mating of closely related individuals, which increases the frequency of harmful recessive alleles. The organisms of Movile Cave have been living in small, isolated populations for millions of years, conditions that would typically be expected to cause significant genetic erosion and inbreeding depression. Yet the ecosystem has persisted, and its constituent species appear to be healthy and functional. How have they avoided the genetic problems that typically plague small, isolated populations? Several mechanisms may be at work. First, the organisms may have purged their most harmful genetic variants through long-term selection. Over millions of years, strongly deleterious alleles that cause severe fitness reductions would be eliminated by natural selection, even in small populations. This process of genetic purging can reduce the severity of inbreeding depression over time, allowing small populations to persist despite their limited genetic diversity. Second, the organisms may have higher mutation rates than surface organisms, providing a steady supply of new genetic variation to compensate for the variation lost through drift. Some studies of cave organisms have found elevated mutation rates, possibly as a consequence of relaxed selection on DNA repair mechanisms in the energy-limited cave environment. However, this remains a hypothesis and has not been directly tested in Movile Cave species. Third, the effective population sizes of the cave species may be larger than they appear. Effective population size is a concept from population genetics that describes the size of an idealized population that would show the same rate of genetic drift as the actual population. In many organisms, the effective population size is much smaller than the census population size due to factors like unequal sex ratios, variation in reproductive success, and overlapping generations. In cave organisms, some of these factors may work in the opposite direction. The long generation times and overlapping generations typical of cave organisms may increase the effective population size relative to the census count, slowing the rate of genetic drift and preserving more genetic variation than would be expected for such small populations. These genetic dynamics are an active area of research, and studies using genomic techniques, whole genome sequencing, population genomic analyses, and functional genomics are beginning to shed light on the genetic health and adaptive capacity of Movile Cave's organisms. As we approach the midpoint of our exploration of Movile Cave, it is worth stepping back and considering the broader significance of this extraordinary place. Movile Cave is not just a biological curiosity, a strange collection of blind, pale animals living in a sulfurous hole in the ground. It is a profound challenge to our understanding of life itself. It demonstrates that complex, self-sustaining ecosystems can exist without sunlight, powered entirely by the chemical energy of the Earth's interior. It shows that evolution in isolation can produce extraordinary levels of specialization and endemic diversity, even in a tiny, confined space. It reveals that life can persist for millions of years under conditions that are, by surface standards, utterly inhospitable: low oxygen, high sulfide, no light, limited energy. These lessons have implications that extend far beyond the borders of a small cave in Romania. They inform our understanding of the origin of life on Earth because the earliest life forms, billions of years ago, likely lived in a world without oxygen, powered by chemical energy sources similar to those in Movile Cave. They inform our search for life on other worlds, because the conditions in Movile Cave resemble the conditions we believe may exist in the subsurface oceans of moons like Europa and Enceladus. And they challenge our philosophical assumptions about the relationship between life and its environment, about what life needs, what it can tolerate, and what it can achieve when left to its own devices in the dark. In the second part of this documentary, we will explore these broader implications in depth. We will examine the parallels between Movile Cave and other extreme ecosystems on Earth. We will consider what the cave can teach us about the search for extraterrestrial life. We will delve deeper into the specific adaptations of the cave's organisms and the molecular mechanisms that allow them to survive in such a hostile environment. And we will reflect on the philosophical and scientific lessons that this remarkable place offers, lessons about the nature of life, the power of evolution, and the hidden richness of the world beneath our feet. But for now, let us leave the cave in its darkness, sealed, silent, patient. Its organisms continue their slow, ancient routines, oblivious to the world above. The bacteria oxidize their sulfide, the amphipods graze their microbial mats, the leeches sense vibrations in the warm, dark water, the spiders listen for the footfall of prey on the cave wall. Life continues as it has continued for 5.5 million years, powered not by the sun but by the slow, steady chemistry of the earth itself. It is a world without light, without weather, without seasons, without change. And yet it is a world that is vibrantly, tenaciously, triumphantly alive. It is Movile Cave, and it has much more to teach us. We left the cave in its silence. We left it sealed beneath the plains of southeastern Romania, its pale inhabitants moving through warm, sulfurous water and stale, oxygen-thin air, indifferent to the human world above. We left it doing what it has done for 5.5 million years, sustaining itself, evolving, enduring, powered by chemistry rather than sunlight, driven by the deep, patient energy of the Earth's interior. In this second part of our journey, we go deeper. We examine the molecular machinery that allows the cave's organisms to survive in conditions that should be lethal. We explore the parallels between Movile Cave and other extreme ecosystems on our planet, from the scalding chimneys of deep-sea hydrothermal vents to the frozen lakes buried beneath Antarctic ice sheets. We consider what the cave's existence tells us about the origin of life on Earth and the possibility of life on other worlds. And we reflect on what it means philosophically, scientifically, and perhaps even spiritually, that such a place exists at all. That in the darkest, most inhospitable corner of our planet, life has not merely survived but has built a world. Let us begin where all biology ultimately begins, at the level of molecules. The survival of organisms in Movile Cave depends on molecular adaptations so precise and so sophisticated that they have taken scientists decades to begin unraveling. The cave's chemically extreme environment, high hydrogen sulfide, low oxygen, elevated carbon dioxide, warm temperatures, constant humidity, imposes stresses on living cells that would be fatal to most organisms without specific biochemical defenses. Understanding how the cave's inhabitants cope with these stresses requires going down to the level of proteins, enzymes, genes, and the fundamental chemistry of life. Hydrogen sulfide is the primary chemical threat in Movile Cave. Its toxicity, as we discussed in Part 1, stems primarily from its ability to inhibit cytochrome C oxidase, the enzyme complex in the mitochondrial inner membrane that catalyzes the final step of aerobic respiration. Cytochrome C oxidase receives electrons from cytochrome C and transfers them to oxygen, reducing it to water. This electron transfer releases energy that is used to pump protons across the mitochondrial membrane, creating the electrochemical gradient that drives the synthesis of ATP, the universal energy currency of living cells. When hydrogen sulfide binds to the iron center in cytochrome C oxidase, it blocks this electron transfer. The enzyme is inhibited. The proton gradient cannot be maintained. ATP synthesis stops. Without ATP, the cell cannot perform any energy-requiring function. It cannot maintain its ion gradients, synthesize proteins, replicate its DNA, or power its molecular motors. It dies. This is the cascade of events that makes hydrogen sulfide lethal to most aerobic organisms, including humans, at concentrations far lower than those found in Movile Cave. The organisms of Movile Cave have found multiple ways to circumvent this problem. The first and most direct approach is modification of cytochrome C oxidase itself. Structural studies of cytochrome C oxidase from sulfide-tolerant organisms have shown that specific amino acid substitutions in the regions surrounding the enzyme's active site can reduce its affinity for hydrogen sulfide without significantly compromising its ability to bind and reduce oxygen. These substitutions change the shape or electrostatic environment of the active site in subtle ways that discriminate against sulfide while maintaining normal catalytic function. Whether the cave's specific organisms have developed such modifications to their cytochrome C oxidase has not been fully established. The technical challenges of obtaining sufficient quantities of purified enzyme from cave organisms for structural studies are enormous. But analogous modifications have been documented in sulfide-tolerant organisms from deep-sea vents and sulfide-rich marine sediments, and it would be surprising if the Movile Cave organisms had not evolved similar solutions to the same problem. A second molecular strategy for coping with hydrogen sulfide is active detoxification, the enzymatic conversion of sulfide to less toxic forms. The key enzyme in this process is sulfide, quinone oxidortictase, or SQR, a membrane-associated enzyme that oxidizes sulfide to elemental sulfur or thiosulfate, donating the extracted electrons to the cell's electron transport chain. This reaction serves two purposes simultaneously. It removes the toxic sulfide before it can inhibit cytochrome C oxidase, and it extracts energy from the oxidation reaction, contributing to the cell's ATP production. SQR has been found in a wide range of organisms, from bacteria to invertebrates to vertebrates. Its presence in animal tissues, including the cells of humans, though at low levels, suggests that sulfide detoxification is an ancient biochemical capability that has been retained throughout the evolution of life, presumably because exposure to geological sulfide has been a recurring challenge throughout biological history. In organisms living in sulfide-rich environments like Movile Cave, SQR activity is expected to be significantly elevated compared to organisms living in sulfide-free environments. Higher levels of SQR expression, driven by the upregulation of the genes encoding the enzyme, would provide more robust sulfide detoxification, allowing the cell to cope with higher concentrations of hydrogen sulfide without suffering inhibition of its respiratory chain. The genes encoding SQR and other sulfide-related enzymes are therefore key targets for molecular studies of cave organism adaptation. By sequencing the genomes of Movile Cave organisms, and comparing the genes involved in sulfide metabolism to those of surface relatives, researchers can identify the specific genetic changes that underlie sulfide tolerance, the mutations and regulatory changes that distinguish a cave organism from its surface ancestor in the most fundamental molecular terms. Such genomic studies have become increasingly feasible with the revolution in DNA sequencing technology that has occurred over the past 2 decades. Next-generation sequencing methods can now produce billions of base pairs of sequence data in a single run, making it possible to sequence entire genomes or transcriptomes, the complete set of genes expressed in a tissue, from small quantities of material. This is crucial for cave research, where the limited access to live animals means that tissue samples must be tiny and precious. Preliminary genomic analyses of several Movile Cave species have been conducted, and while the results are still being processed and analyzed, they are beginning to reveal the molecular signatures of adaptation to the cave environment. These studies look for positive selection, the accelerated evolution of specific genes driven by natural selection, in genes related to sulfide metabolism, oxygen sensing, sensory systems, eye development, and pigmentation pathways. Genes under positive selection show higher rates of amino acid changing mutations compared to synonymous mutations, a signature that indicates the mutations are being driven by selection rather than occurring randomly. Early results suggest that genes involved in detoxification, antioxidant defense, and sensory function show evidence of positive selection in Movile Cave organisms, exactly what would be expected if the cave environment has been a powerful driver of adaptive evolution in these gene categories. Meanwhile, genes involved in eye development and pigmentation show signatures of relaxed selection, elevated rates of potentially deleterious mutations accumulating without being removed by selection, consistent with the functional degeneration of these systems in the cave environment. Beyond hydrogen sulfide tolerance, the organisms of Movile Cave face the challenge of functioning at very low oxygen concentrations. The atmospheric oxygen level in the cave, 7 to 10%, is roughly one-third to one-half the surface level of 21%. In the water, where oxygen must dissolve from this already thin atmosphere, concentrations are even lower. For aerobic organisms that depend on oxygen for respiration, this hypoxia, oxygen deficiency, is a constant physiological challenge. Aerobic organisms cope with hypoxia through a cascade of molecular responses orchestrated by a family of transcription factors known as hypoxia-inducible factors, or HIFs. When oxygen levels drop, HIFs are activated and enter the cell nucleus, where they bind to specific DNA sequences and alter the expression of hundreds of target genes. These gene expression changes adjust the cell's metabolism to cope with reduced oxygen availability, increasing the production of oxygen-binding proteins, stimulating the growth of new blood vessels or equivalent oxygen delivery structures, shifting metabolism toward glycolysis, which does not require oxygen, and reducing energy-consuming activities to match the reduced energy production capacity. The HIF system is ancient and highly conserved across the animal kingdom, from simple invertebrates to complex vertebrates. It is the universal molecular response to oxygen deficiency, and it is almost certainly operating in the animals of Movile Cave. But chronic exposure to hypoxia, not just occasional low oxygen episodes but permanent, lifelong generational exposure, likely drives additional adaptations beyond what the HIF response alone can achieve. One such adaptation is changes in the oxygen-binding proteins themselves. Hemoglobin in vertebrates and the analogous hemocyanin or myoglobin in many invertebrates is responsible for binding oxygen in the tissues where it is most concentrated and releasing it where it is needed. The oxygen affinity of these proteins, how tightly they bind oxygen, can be tuned by amino acid changes in their structure. An oxygen-binding protein with a higher affinity for oxygen can extract oxygen from environments where the concentration is very low, allowing the organism to maintain adequate oxygen delivery to its tissues even when environmental oxygen is scarce. The leeches and crustaceans of Movile Cave likely have hemoglobin or similar proteins with elevated oxygen affinity, modified by natural selection over millions of years to extract the maximum possible oxygen from the cave's thin, oxygen-poor atmosphere. While direct measurements of oxygen binding curves for Movile Cave organisms have not been published, comparisons with other cave-adapted and hypoxia-adapted organisms suggest this is a highly probable adaptation. The mitochondria themselves, the organelles responsible for aerobic respiration, may also be modified in Movile Cave organisms. Mitochondrial efficiency, measured as the amount of ATP produced per oxygen molecule consumed, can vary considerably depending on the composition of the electron transport chain proteins. Organisms adapted to chronic hypoxia in other contexts, high-altitude animals, deep-diving marine mammals, have been found to have more efficient mitochondria than their lowland or non-diving relatives, extracting more energy from each oxygen molecule and thereby reducing the total oxygen consumption needed to maintain a given energy output. If the Movile Cave organisms have evolved more efficient mitochondria through similar mechanisms, they could potentially sustain adequate ATP production at oxygen concentrations that would leave unadapted organisms in a state of severe energy deficit. This would represent an elegant molecular solution to the problem of chronic hypoxia, not by reducing the need for energy, but by extracting energy more efficiently from the limited oxygen available. The cave's elevated carbon dioxide levels present yet another molecular challenge. Carbon dioxide, when dissolved in water, forms carbonic acid, which dissociates to produce bicarbonate and hydrogen ions. This lowers the pH, makes the solution more acidic, which in turn affects the function of proteins, particularly enzymes, whose activity is highly sensitive to pH. High internal carbon dioxide concentrations also affect the behavior of hemoglobin and similar oxygen-carrying molecules, reducing their oxygen affinity through a phenomenon known as the Boer effect. For organisms living in Movile Cave's high carbon dioxide environment, maintaining stable intracellular pH despite elevated carbon dioxide is crucial. The primary mechanism for managing this challenge is carbonic anhydrase, an enzyme that catalyzes the rapid interconversion of carbon dioxide, water, bicarbonate, and protons. By controlling the distribution of these species across biological membranes, carbonic anhydrase helps regulate intracellular pH and facilitate the excretion of excess carbon dioxide from tissues. Organisms in carbon dioxide-rich environments typically have elevated levels of carbonic anhydrase activity, and the genes encoding this enzyme may show signatures of positive selection, indicating that natural selection has acted to optimize carbonic anhydrase function for the specific pH and carbon dioxide conditions of the cave. The Movile Cave organisms, having experienced elevated carbon dioxide concentrations for millions of generations, are likely to have evolved particularly effective carbonic anhydrase systems, though this hypothesis awaits direct experimental confirmation. The cumulative picture emerging from these molecular considerations is one of extraordinary complexity and integration. Surviving in Movile Cave is not a matter of having one or two specific adaptations. It requires a coordinated suite of molecular changes across multiple biochemical systems: sulfide detoxification, oxygen utilization, pH regulation, sensory system enhancement, and many more. These changes must work together coherently. They must be compatible with one another and with the overall functioning of the organism's biochemistry. The evolution of each adaptation must not compromise the others. This requirement for integrated, compatible adaptations is one of the reasons why evolution in extreme environments takes so long. It is relatively easy for evolution to make a single biochemical change, a mutation in one gene that confers some advantage in a challenging environment. It is much harder to make dozens or hundreds of compatible changes simultaneously, each contributing to the organism's fitness in the new environment without disrupting the function of other biochemical pathways. This process takes millions of years and countless generations of selection, which is precisely what Movile Cave has provided. The microbial dimension of the cave's biology continues to yield new discoveries as molecular techniques become more powerful and more sensitive. Metagenomic studies, analyses of DNA extracted directly from cave samples without prior cultivation, have revealed a microbial world of staggering complexity, far beyond what was initially appreciated when the cave was first explored with traditional microbiological methods. Traditional microbiology relies on the cultivation of organisms, growing them in the laboratory on defined growth media, and then studying the resulting colonies. This approach has severe limitations because most microorganisms cannot be cultivated under laboratory conditions. They may require specific chemical substrates, growth factors, or environmental conditions that are difficult or impossible to replicate in the lab. Estimates suggest that only 1 to 10% of microbial species in any environmental sample can be cultured under standard laboratory conditions. The other 90 to 99%, the so-called uncultured majority, remain invisible to traditional methods. Metagenomics bypasses this limitation by sequencing all of the DNA present in an environmental sample simultaneously, regardless of whether the organisms it came from can be cultivated. By assembling the resulting sequence data into genomic fragments and comparing these fragments to databases of known gene sequences, researchers can identify the types of organisms present, infer their metabolic capabilities, and reconstruct their ecological roles, all without ever growing a single cell in the laboratory. Metagenomic analyses of Movile Cave samples have revealed dozens of previously unknown microbial lineages, organisms so different from anything described before that they cannot be classified within existing taxonomic groups. These novel organisms possess genes encoding metabolic enzymes that suggest they are performing chemical reactions not previously known to occur in cave environments, novel pathways for energy extraction, novel mechanisms for carbon fixation, novel strategies for dealing with the cave's toxic chemicals. One particularly intriguing finding from metagenomic studies is the presence of organisms with the potential to perform anaerobic methane oxidation, the oxidation of methane using sulfate as an electron acceptor rather than oxygen. This process, which is carried out by a consortium of archaea and bacteria, is well established in marine sediments, where it plays an important role in preventing methane, a potent greenhouse gas, from reaching the atmosphere. In Movile Cave, anaerobic methane oxidation could represent an additional pathway of carbon cycling, converting methane to bicarbonate and coupling this process to the reduction of sulfate to sulfide. If this process is occurring in Movile Cave, it would create an interesting metabolic coupling. Hydrogen sulfide rising from the deep thermal waters fuels aerobic chemosynthesis, which produces sulfate. The sulfate is reduced back to sulfide by anaerobic sulfate reducers, The regenerated sulfide can be oxidized again by aerobic chemosynthesizers, while methane is oxidized anaerobically using the sulfate produced by this cycle. This would create a tightly coupled, internally self-sustaining biogeochemical cycle, a microbial perpetual motion machine, continuously cycling sulfur and carbon through multiple oxidation states using the energy of the incoming thermal waters to drive the whole system. Such tight metabolic coupling between different microbial groups is a common feature of extreme ecosystems, and it represents one of the ways in which microbial communities can sustain themselves in environments where energy is scarce. By linking the waste products of one metabolic process to the inputs of another, different microbial species create a network of interdependence that maximizes the efficiency with which the available energy is utilized. Nothing is wasted. Every chemical byproduct becomes someone else's fuel. The broader context of extreme ecosystems on Earth provides important perspective for understanding Movile Cave. The cave is not unique in being an ecosystem powered by chemosynthesis. It is unique in the specific combination of features it possesses. Understanding where it fits in the global landscape of extreme and chemosynthetic ecosystems illuminates what makes it special and what makes it representative of a broader biological phenomenon. Deep-sea hydrothermal vents are perhaps the most famous chemosynthetic ecosystems, certainly the ones that have received the most scientific attention since their discovery in 1977. Hydrothermal vents form at mid-ocean ridges, where tectonic plates are pulling apart and magma wells up from the mantle to fill the gap. Seawater percolates down through fractures in the ocean floor, is heated to temperatures of several hundred degrees Celsius by the underlying magma, dissolves minerals and gases including hydrogen sulfide and carbon dioxide from the hot rock, and then rises back to the seafloor as a superheated, mineral-rich fluid. Where this fluid meets the cold, oxygenated deep ocean water, it precipitates minerals, building the towering chimney structures that give hydrothermal vents their dramatic appearance, and creates sharp chemical gradients that chemosynthetic bacteria exploit to generate energy. These bacteria form the base of a remarkably rich food web, supporting communities of tube worms, clams, shrimp, crabs, fish, and many other organisms at some of the most productive areas of the deep ocean floor. The parallels with Movile Cave are immediately apparent. Both are dark, chemosynthetic ecosystems based on hydrogen sulfide oxidation. Both feature symbioses between animals and chemosynthetic bacteria. Both harbor unique, specially adapted species. But there are also important differences. Hydrothermal vents are geologically ephemeral. Individual vent fields last only decades to centuries before the magmatic heat source wanes and the vent goes cold. The Movile Cave ecosystem, by contrast, has persisted for millions of years. Vent communities must therefore be capable of rapid colonization and rapid dispersal, strategies that are the opposite of the slow, isolated evolution characteristic of Movile Cave. Cold seeps are another type of marine chemosynthetic ecosystem, found in areas of the ocean floor where cold, hydrocarbon-rich fluids seep from below, often associated with natural gas deposits or the decomposition of gas hydrates in the seafloor sediments. Like vents, cold seeps support chemosynthetic communities based on methane oxidation and sulfate reduction. Cold seeps are generally longer-lived than hydrothermal vents. They can persist for thousands to tens of thousands of years, and their communities show higher levels of endemism and specialization, reflecting the longer period of isolation and adaptation. Subglacial lakes, lakes of liquid water trapped beneath thick ice sheets, like the famous Lake Vostok beneath the East Antarctic ice sheet, represent yet another type of extreme, isolated chemosynthetic ecosystem. These lakes have been sealed from the atmosphere by overlying ice for millions of years, cut off from sunlight and from surface organic matter inputs. Any life that exists in these lakes must be sustained by chemosynthesis, drawing on minerals dissolved from the underlying bedrock by the slowly circulating water. Recent drilling projects have confirmed that Lake Vostok and other subglacial Antarctic lakes harbor microbial life, bacteria and archaea adapted to the cold, dark, nutrient-poor conditions. The similarity to Movile Cave is striking. Both are sealed, dark, isolated systems where chemosynthesis sustains microbial communities under extreme conditions. The subglacial lakes are even more isolated than Movile Cave in some respects. They are separated from the surface not by clay and rock, but by 4 kilometers of ice, a barrier that has been even more effective at preventing exchange with the surface. Terrestrial hot springs and geothermal areas are another relevant comparison. In places like Yellowstone or the geothermal fields of Iceland and New Zealand, thermal waters rich in hydrogen sulfide, methane, and other reduced chemicals support communities of thermophilic and chemosynthetic microorganisms. While these environments are not sealed, they are in contact with the atmosphere and receive inputs from the surface. They demonstrate the same fundamental principle as Movile Cave, that geological chemical energy can sustain rich microbial communities wherever the right chemical conditions exist. What emerges from this global survey of extreme ecosystems is a picture of life as an opportunistic, flexible, resilient phenomenon, one that has evolved to exploit every available energy source in every available environment, from the scalding temperatures of hydrothermal vents to the freezing darkness of subglacial lakes, from the sulfurous chambers of Movile Cave to the hydrocarbon-saturated sediments of cold seeps. Wherever there is chemical energy and liquid water, life has found a way. This conclusion, wherever there is chemical energy and liquid water, life has found a way, is the foundation of astrobiology, the scientific discipline that seeks to understand the distribution and nature of life in the universe. And it is precisely this conclusion that makes Movile Cave so relevant to the search for life on other worlds. For much of the history of the search for extraterrestrial life, scientists focused on planets and moons that received adequate sunlight to support photosynthesis, worlds in the so-called habitable zone, the range of distances from a star where liquid water can exist on the surface. This search was implicitly biased toward surface life, toward photosynthesis, toward the particular form of life that evolved on Earth's sunlit surface. It was a search for worlds like ours. The discovery of chemosynthetic ecosystems at hydrothermal vents in subglacial lakes and in sealed caves like Movile Cave has fundamentally changed this perspective. If complex ecosystems can sustain themselves without sunlight, powered entirely by geological and chemical energy, then the habitable zone concept needs to be radically expanded. Life does not need to be on the surface of a planet. It does not need to receive sunlight. It does not need a clement, Earth-like environment. It needs liquid water and chemical energy, conditions that may be far more common in the universe than previously appreciated. The moons of the outer solar system are the most immediate beneficiaries of this expanded perspective. Europa, the fourth largest moon of Jupiter, is covered by a global ocean of liquid water beneath a shell of ice several kilometers thick. This ocean exists not because Europa receives much solar energy. It orbits Jupiter at 5 times Earth's distance from the Sun, where solar energy is 25 times weaker than at Earth, but because Jupiter's powerful gravitational forces flex and squeeze Europa's rocky interior, generating heat through tidal friction. This tidal heating keeps Europa's interior warm enough to maintain a liquid water ocean even at its extreme distance from the Sun. The floor of Europa's ocean, where it contacts the rocky mantle beneath, is likely to be geothermally active, heated by the same tidal forces that maintain the liquid ocean above. Where warm, geothermally heated water contacts the ocean floor, chemical reactions between the hot rock and the cold water would produce hydrogen sulfide, hydrogen, and other reduced compounds, the same chemical substrates that fuel chemosynthetic ecosystems on Earth. If hydrothermal vents exist on Europa's ocean floor, chemosynthetic microbial communities similar to those at Earth's own hydrothermal vents might thrive there. Enceladus, a small moon of Saturn, provides even more compelling evidence. In 2006, the Cassini spacecraft discovered that Enceladus is actively venting plumes of water vapor and ice particles from fissures near its south pole, the so-called tiger stripes. Subsequent analysis of these plumes revealed the presence of water, salt, silica nanoparticles, and crucially molecular hydrogen, methane, and organic compounds. The presence of molecular hydrogen is particularly significant because it indicates that hot water is reacting with rocky minerals at depth through a process called serpentinization, the same process that produces hydrogen at some of Earth's seafloor hydrothermal systems. The molecular hydrogen produced by serpentinization can serve as an energy source for microorganisms that reduce carbon dioxide to methane, methanogens, a group of archaea that are among the most ancient forms of life on Earth. If methanogens exist on Enceladus, they would represent the first confirmed example of extraterrestrial life, living in a warm, hydrogen-rich, chemically energized ocean beneath the moon's icy crust. Titan, another moon of Saturn, presents different but equally intriguing possibilities. Titan has a thick atmosphere and seas and lakes on its surface, but not of water. Titan is so cold, minus 179 degrees Celsius, that water is frozen solid. Instead, its lakes and seas are composed of liquid methane and ethane. Could life exist in liquid methane, using chemical energy sources to power its metabolism without oxygen? This is a more speculative possibility, requiring forms of biochemistry radically different from anything on Earth, but the question itself is a direct product of the paradigm shift enabled by the discovery of chemosynthetic ecosystems like those in Movile Cave. Movile Cave has thus become a touchstone for astrobiologists, a terrestrial analog for the kinds of chemosynthetic ecosystems that might exist in the subsurface oceans of icy moons. It demonstrates that such ecosystems can persist for geological timescales, that they can support genuine biological complexity beyond mere microbial films, and that they can do so in conditions, low oxygen, high chemical toxins, no light, steady-state energy supply, that broadly resemble what we might expect in an icy moon's subsurface ocean. The comparison is not perfect, of course. Europa's putative ocean is saltwater, deep, cold, and dark throughout, not warm and humid like Movile Cave. The specific chemistry differs, the geological context differs, but the underlying principle is the same. Chemical energy plus liquid water can sustain life in the absence of sunlight, and that life can, over geological time, develop surprising complexity and diversity. The astrobiological significance of Movile Cave also extends to questions about the origin of life itself, not on other worlds but here on Earth, billions of years ago. The very first living organisms on Earth arose in conditions radically different from today's surface environment, and understanding those conditions is crucial to understanding how life began. The early Earth, 4 billion years ago, was a hostile and dynamic place. The atmosphere contained almost no oxygen. It was composed primarily of nitrogen, carbon dioxide, methane, hydrogen sulfide, and water vapor. There was no ozone layer to block ultraviolet radiation, which bombarded the surface continuously. Volcanic activity was far more intense than today, covering vast areas with lava and injecting enormous quantities of gases into the atmosphere and oceans. The oceans were warm and acidic, rich in dissolved minerals from the intense volcanic activity. In this environment, photosynthesis did not yet exist. The sophisticated molecular machinery of photosynthesis, the chlorophyll, the reaction centers, the carbon fixation enzymes had not yet evolved. Life, in its earliest form, could not have been photosynthetic. It must have been chemosynthetic, powered by chemical energy rather than light. The leading hypotheses for the origin of life on Earth invoke chemical environments very similar to those in Movile Cave. Hydrothermal vent systems, both seafloor vents and terrestrial hot springs, have been proposed as the cradle of life, the settings where the first self-replicating molecules emerged from complex mixtures of organic chemicals. These environments feature the key requirements: liquid water, chemical energy gradients, particularly involving hydrogen sulfide and hydrogen, mineral surfaces that could catalyze chemical reactions, and a steady supply of the raw chemical ingredients for building organic molecules. The chemosynthetic ecosystem of Movile Cave, while obviously far more complex than anything that existed 4 billion years ago, embodies the same fundamental principles that are thought to have governed the chemistry of life's origin. Hydrogen sulfide as an energy source, carbon dioxide as a carbon source, chemical gradients driving the flow of energy through biological systems, no sunlight required. There is a hypothesis in origins of life research known as the alkaline hydrothermal vent hypothesis, proposed by Michael Russell and colleagues, which argues that life arose at hydrothermal vents where alkaline, hydrogen-rich fluids from the Earth's mantle met the acidic, carbon dioxide-rich early ocean. These vents would have created natural proton gradients, the same kind of gradient that modern cells use to synthesize ATP, across mineral membranes in the vent chimneys, providing the first source of cellular energy. In this framework, the earliest protocells were not cells in the modern sense. They did not have lipid membranes or complex proteins. They were simply mineral compartments, pores and chambers in the hydrothermal vent minerals, where concentration and chemical gradients could drive the synthesis of simple organic molecules. Over billions of years, these protocells acquired increasingly complex molecular machinery, eventually developing true cell membranes, genetic information systems, and the sophisticated enzyme networks of modern biochemistry. If this hypothesis is correct, then chemosynthesis predates photosynthesis by billions of years. Life began in the dark, powered by geological chemical energy, in conditions not entirely unlike those in Movile Cave. Photosynthesis was a later invention, a revolutionary biochemical innovation that allowed life to tap into the far more abundant energy of sunlight and thereby expand across the sunlit surface of the planet. From this perspective, Movile Cave is not just a curiosity, it is a glimpse into the deep past. It is a preserved example of the kind of dark, chemosynthetic world in which life began. Its bacteria, oxidizing sulfide and methane and ammonium in total darkness, are not primitive. They are sophisticated organisms with billions of years of evolution behind them. But their way of life, chemosynthetic, oxygen-independent, driven by geological chemical energy, echoes the first biology that ever existed on Earth. The study of Movile Cave has generated not only scientific insights but methodological innovations, new approaches to investigating biology in extreme, inaccessible environments that have found applications far beyond the cave itself. One such innovation is the development of minimally invasive sampling protocols, methods for collecting biological samples from the cave without significantly disturbing the ecosystem. Traditional field sampling methods, netting animals, scraping surfaces, collecting water, are too disruptive for use in an ecosystem as fragile as Movile Caves. Researchers have developed specialized micro-sampling tools, sterile sampling chambers, and remote sensing devices that can collect nanogram-scale tissue samples, microliter volumes of water, and microscale rock scrapings without leaving a significant footprint in the cave. These protocols have been adapted for use in other sensitive environments: pristine glacier cavities, deep aquifer systems, pharmaceutical manufacturing clean rooms, and even extraterrestrial sample collection simulations, because the fundamental challenge is the same: how to collect representative, uncontaminated samples from an environment that must not be disturbed. Another methodological innovation is the development of in situ analytical techniques, measurements made directly in the cave rather than on samples brought back to the laboratory. Many of the cave's organisms and chemical species are so unstable that they change rapidly after removal from the cave environment. Gas compositions, dissolved chemical concentrations, and even some organismal behaviors are best measured in real time, inside the cave, using portable analytical instruments that can be brought through the access shaft. Researchers working in Movile Cave have developed or adapted portable versions of gas chromatographs, dissolved oxygen sensors, electrochemical sulfide analyzers, and pH meters for use in the cave's cramped, humid, chemically hostile conditions. These instruments must be waterproof, sulfide-resistant, battery-operated, and small enough to be carried through passages that sometimes require researchers to crawl. The engineering challenges are considerable, but the resulting instruments have provided data of a quality and specificity that would be impossible to obtain from samples taken out of the cave. These methodological developments have implications for future exploration of extreme environments both on Earth and potentially on other worlds. The instruments and protocols developed for Movile Cave research are directly relevant to the challenge of characterizing subsurface environments on Mars, where drilling is required to reach potentially habitable zones, and on icy moons like Europa and Enceladus, where any life that exists would likely be in a sealed, chemically extreme subsurface ocean. The lessons learned from working in Movile Cave, how to sample without contaminating, how to measure without disturbing, how to infer complexity from minimal data, are lessons that space exploration will need to apply. The question of contamination is particularly important, both for protecting Movile Cave and for drawing lessons relevant to planetary exploration. The concept of planetary protection, preventing Earth organisms from contaminating other worlds, and preventing any potential extraterrestrial organisms from contaminating Earth, is a central concern of space exploration. The protocols developed for Movile Cave, where the concern is preventing surface organisms from contaminating the cave's pristine ecosystem, are a useful terrestrial analog for the challenges of planetary protection. Every human who enters Movile Cave brings billions of microorganisms with them, on their skin, in their respiratory system, on their equipment. Even after rigorous sterilization of clothing and equipment, it is essentially impossible to eliminate all microbial contamination. Researchers must therefore limit their visits, minimize physical contact with cave surfaces, and monitor the cave's microbial community over time to detect any signs of contamination. The monitoring program for Movile Cave uses molecular methods, periodic sampling of the cave's microbial communities in comparison to a baseline established from the earliest, least contaminated samples. By comparing the gene sequences of microorganisms in current samples to those in baseline samples, researchers can detect the presence of surface-derived microorganisms and assess whether they are establishing themselves in the cave or simply dying out. So far, the evidence suggests that the cave's native microbial community has remained largely intact, with no permanent establishment of foreign species, a testament to the effectiveness of the protective protocols and perhaps to the competitive superiority of the cave's well-adapted native organisms over any foreign invaders. This monitoring approach is directly relevant to planetary protection. When spacecraft are sent to potentially habitable worlds, they must be sterilized to reduce the risk of contaminating those worlds with Earth life. But sterilization is never perfect. Some microorganisms, particularly the hardy bacterial endospores of species like Bacillus subtilis, can survive conventional sterilization protocols and potentially survive the journey through space. Understanding how foreign microorganisms behave when introduced into an extreme, well-adapted ecosystem, whether they die out, survive transiently, or establish permanently, is crucial information for assessing the risks of planetary contamination. Movile Cave provides a natural experiment in this question. The cave has been receiving minute quantities of surface microorganisms with every human visitor since its discovery in 1986. The fact that its native community has remained stable suggests that the cave's ecological conditions, low oxygen, high sulfide, limited nutrients, intense microbial competition, are effective barriers to colonization by surface organisms. This is encouraging news for planetary protection. Extreme environments on other worlds may be naturally resistant to colonization by Earth organisms, even if small numbers survive the journey. The philosophical dimensions of Movile Cave deserve attention, because this place raises profound questions about the nature of life, the place of consciousness in the universe, and the relationship between the visible world and the hidden worlds that exist beyond our direct perception. Consider what it means that there is a complete self-sustaining world, an ecosystem with producers, consumers, predators, and decomposers, operating beneath our feet without our knowledge or participation. For the entire history of human civilization, the construction of pyramids, the rise and fall of empires, the development of agriculture, the discovery of fire, this cave has been ticking along in the dark, unchanged and unaware of us. Its leeches have hunted their amphipods, its spiders have stalked their springtails, its bacteria have oxidized their sulfide. The whole intricate clockwork of the ecosystem has continued without pause, without knowledge of the sun, without any connection to the human world above. This is a humbling thought. We tend to think of ourselves as the central actors in the story of life on Earth, the species that has named all the others, that has mapped and measured and cataloged the biological world. But Movile Cave existed for 5.5 million years before we found it. It will almost certainly exist for millions of years after we are gone. Its organisms were doing their complex, sophisticated, highly evolved thing long before our species appeared, and they will continue long after we disappear. They are not supporting players in our story. If anything, we are the latecomers, the brief visitors who punched a hole in the roof of their world and peeked in. There is also something profound about the absolute darkness of the cave, the fact that nothing in it has ever been seen. Not in any visual sense, not by any organism capable of vision. The microbial mats have never been lit by anything other than the scientist's flashlight. The spiders have never seen their own webs. The leeches have never perceived the outline of their prey. All of the complexity and beauty of this ecosystem, the intricate architecture of the microbial mats, the elegant adaptations of the cave's animals, the chemical dance of the food web exists entirely unwitnessed, in perfect darkness, beyond the reach of any visual perception. This is not a tragic observation. The cave's organisms do not need to be seen to be real, to be important, to be alive. Their existence does not depend on our observation of them. But it does challenge our tendency to equate visibility with reality, to assume that the world is essentially what we can see and measure. Movile Cave reminds us that enormous complexity can exist in total darkness, unperceived and unsuspected for geological timescales. How many other such worlds exist, on Earth or elsewhere, beyond the reach of our senses and instruments? The question of what we do not know, what we cannot yet perceive, is perhaps the most important question in science. The history of scientific discovery is largely a history of expanding perception. The telescope revealing the vastness of the universe, the microscope revealing the invisible world of cells and microorganisms, molecular biology revealing the information content of DNA, and now astrobiology beginning to reveal the possibility of life beyond Earth. Each expansion of our perceptual range reveals new worlds, new levels of complexity, new forms of existence, new answers to the question of what is possible. Movile Cave is one such revelation. It expanded our perceptual range in a crucial direction, downward into the dark, into the hidden chemical world of the subsurface. It showed us that our categories, photosynthesis as the foundation of complex life, sunlight as the prerequisite for biological productivity, the surface environment as the defining context for evolution, were too narrow. It showed us that life is more creative, more flexible, and more resourceful than we had imagined. The implications for the search for extraterrestrial life extend beyond the simple observation that life can exist without sunlight. Movile Cave and the broader class of chemosynthetic ecosystems raise deeper questions about the detectability of life on other worlds. If life on Europa or Enceladus exists in a sealed subsurface ocean, how would we detect it? The organisms would leave no visible signature on the surface of the moon. There would be no green coloration from photosynthetic pigments, no atmospheric oxygen from the splitting of water, no large-scale modification of the surface landscape. The life would be as invisible to remote observation as Movile Cave is from the surface of Romania, present, complex, and active, but completely hidden beneath a barrier of ice or rock. The best hope for detection would be through the very channels by which the cave's existence was revealed, geochemical signals rising from the subsurface to the accessible environment. In the case of Enceladus, the plumes erupting from the South Pole offer exactly this opportunity. They carry material from the subsurface ocean directly into space, where a spacecraft can sample it without needing to drill through the ice. The detection of molecular hydrogen, methane, and complex organic molecules in the Cassini plume data suggests that the subsurface ocean has the chemistry to support life, but detecting actual biological molecules, specific amino acids, lipids, or nucleotides that could only be produced by biological processes, would require more sophisticated chemical analysis than Cassini was equipped to perform. Future missions to Enceladus and to Europa, where similar but smaller plumes may exist, are being designed with this goal in mind. Instruments capable of detecting and characterizing complex organic molecules at extremely low concentrations, distinguishing biological from abiotic chemistry, and identifying the isotopic signatures of biological processes could potentially detect life in these extraterrestrial oceans without ever landing on the ice surface. But the lessons of Movile Cave also suggest that the absence of such signals should not be taken as evidence of absent life. The cave is chemically active. It produces hydrogen sulfide, methane, and other gases that rise toward the surface, but these geochemical signals are so small that they would be undetectable from space or even from the surface above the cave. The most sensitive remote sensing instruments available today would not detect any biological signal from Movile Cave if they were scanning the Dobrogea plain from above. If the cave had not been discovered accidentally through a borehole, it would remain completely unknown. This suggests a profound detection problem for astrobiology. Chemosynthetic life, sealed in a subsurface environment, may leave no detectable surface signature. It may produce no oxygen, no visible pigments, no measurable temperature anomaly, no spectroscopic feature that a telescope could detect. It could exist in complete secrecy, as Movile Cave existed for 5.5 million years, visible only to instruments that physically penetrate the barrier between the surface and the subsurface. This means that the question, "Is there life on Europa?" may not be answerable by remote sensing alone. It may require drilling through the ice, sampling the ocean, and bringing the ocean directly to sophisticated analytical instruments. This is an enormous technical and logistical challenge. The ice shell on Europa is estimated to be 10 to 30 kilometers thick, but it is not an insuperable one. Future generations of space exploration technology may make it achievable. And if it is achieved, and if life is found, the discovery will owe something to the tiny, sealed cave in Romania that first demonstrated, beyond any reasonable doubt, that complex life can sustain itself without sunlight in a chemically extreme, isolated subsurface environment. Returning to Earth, and to the immediate practical implications of Movile Cave research, the cave's organisms have attracted considerable interest from biotechnology and medicine. Organisms adapted to extreme environments, extremophiles, often produce enzymes and other biological molecules with unusual properties that make them useful for industrial or medical applications. The chemosynthetic bacteria of Movile Cave, adapted to function in high sulfide, low oxygen, and warm temperatures, likely produce a range of novel enzymes, particularly enzymes involved in sulfur metabolism, oxidation reactions, and toxin handling, that might have applications in bioremediation, the treatment of sulfide-contaminated industrial wastewaters, or the development of new pharmaceutical compounds. The detoxification enzymes that allow the cave's bacteria to metabolize hydrogen sulfide efficiently could potentially be harnessed to develop biosystems for removing sulfide from industrial effluents, a significant environmental problem in the mining, petroleum refining, and paper manufacturing industries. Hydrogen sulfide is, ironically, not only a toxic pollutant in industrial contexts, but also a signaling molecule in biological tissues, a so-called gas transmitter, similar in some respects to nitric oxide. Research over the past 2 decades has revealed that small, tightly regulated concentrations of hydrogen sulfide in mammalian tissues play important physiological roles in vascular regulation, neurotransmission, cytoprotection, and anti-inflammatory responses. Enzymes that regulate hydrogen sulfide metabolism, producing it in precisely controlled quantities or degrading it before it reaches toxic concentrations, are thus of significant medical interest. The sulfide metabolizing enzymes of Movile Cave organisms, shaped by millions of years of evolution in a high sulfide environment, may represent some of the most sophisticated and efficient sulfide handling enzymes in the biological world. Understanding their structure, function, and regulation could provide insights into how biological systems manage hydrogen sulfide at the molecular level, with potential applications in the treatment of cardiovascular disease, neurodegenerative disorders, and inflammatory conditions, all of which involve dysregulation of hydrogen sulfide signaling. The cave's organisms may also be sources of novel antibiotics. The intense competition for limited resources in the cave ecosystem is a powerful driver of the evolution of antimicrobial compounds, chemicals that bacteria produce to inhibit or kill competing bacteria. In the microbial mats of Movile Cave, where dozens of microbial species compete for the same chemical substrates in a confined space, the arms race between competing microbial populations may have driven the evolution of particularly potent and novel antimicrobial compounds. Many of the most important antibiotics in clinical use today, penicillin, streptomycin, erythromycin, were first isolated from bacteria living in environments of intense microbial competition, particularly soils. The Movile Cave microbial community may be an analogous source of novel antibacterial compounds adapted to fight the specific bacterial lineages present in the cave, lineages that may be phylogenetically distant from the pathogens that current antibiotics were designed to combat, and therefore potentially effective against drug-resistant strains. The conservation status of Movile Cave deserves careful consideration. The cave is protected by Romanian law and international agreements, and access is strictly controlled, but protection from human visitation is not the same as protection from all threats. The greatest long-term threat to Movile Cave may be changes in the geological and hydrological systems that sustain it. The thermal waters that bring chemical energy to the cave originate from a deep aquifer system that is connected, however distantly, to the surface hydrological network. Changes in surface land use, groundwater extraction, or climate-driven changes in precipitation patterns could potentially alter the flow rate, temperature, or chemical composition of these thermal waters, with cascading effects on the cave ecosystem. Industrial activity in the vicinity of the cave poses another potential threat. The Dobrogea region has historically been an area of agricultural and industrial activity, including petroleum extraction. Contamination of groundwater by agricultural chemicals, petroleum byproducts, or industrial effluents could infiltrate the aquifer system feeding the cave, introducing foreign chemicals that the cave's organisms are not adapted to handle. Even small concentrations of pesticides, petroleum hydrocarbons, or heavy metals could disrupt the cave's finely balanced microbial chemistry, with potentially devastating effects on the ecosystem. Climate change is a more diffuse but equally real threat. While the cave's internal temperature is primarily determined by the geothermal waters rather than the surface climate, changes in surface temperature and precipitation patterns could indirectly affect the cave by altering groundwater recharge rates, changing the dissolved mineral content of infiltrating water, or modifying the depth of permafrost, which, while not currently present in this relatively warm region, could become relevant under extreme climate scenarios. The scientific community has advocated strongly for maintaining and strengthening the protective measures around Movile Cave, including establishing a larger buffer zone around the cave perimeter to prevent incompatible land uses, implementing regular monitoring of groundwater quality in the surrounding area, and developing protocols for rapid response to any detected threats to the cave ecosystem. These conservation efforts are important not just for the sake of the cave's unique biological heritage, but for the scientific value of the cave as an ongoing natural experiment. 5.5 million years of evolution cannot be replicated or recreated. If the Movile Cave ecosystem is disrupted or destroyed, that experiment is over. The knowledge that could have been gained from centuries of future research, knowledge about evolution, about extreme life, about the foundations of chemosynthesis, about the origins of life, is lost forever. This is a genuinely irreplaceable natural resource. There is no other place on Earth quite like Movile Cave. No other terrestrial chemosynthetic ecosystem has been sealed from the surface for this long, has developed this degree of endemic biodiversity, and has been this thoroughly studied. It is a window into a hidden world, and like all windows, it can be broken. As we deepen our understanding of the cave's biology through molecular and ecological studies, new questions continually emerge. One of the most intriguing is the question of how the cave's organisms manage the transition from the surface to the cave environment over evolutionary time. We know that the cave's organisms descended from surface-dwelling ancestors that entered the cave before it was sealed, approximately 5.5 million years ago. But the cave's current conditions, high sulfide, low oxygen, complete darkness, are extreme. The surface ancestors would not have been adapted to these conditions. They would have entered the cave as ordinary surface organisms and would have faced an environment that was, at best, highly stressful and at worst, lethal. How did they survive long enough for evolution to adapt them? This is the cave colonization paradox, the challenge of explaining how organisms could have initially tolerated conditions that their descendants are now exquisitely adapted to, but that would presumably have been toxic or physiologically challenging for the unadapted colonizers. Several possibilities have been proposed. First, the cave's conditions at the time of colonization may have been less extreme than they are today. If the thermal waters initially flowing into the cave were less sulfide-rich, or if the cave's atmosphere initially had more connection to the surface and therefore higher oxygen, the conditions at the time of colonization might have been tolerable for surface organisms. Over time, as the cave became more sealed and the water chemistry changed, conditions became more extreme, and the organisms within were forced to adapt or die. Second, the colonizing organisms may not have been typical surface organisms, but rather species that were already living in moderately stressful environments, sulfide-containing springs, oxygen-depleted swamps, dark soil environments, and that therefore already had some degree of pre-adaptation to the cave's conditions. Such organisms, entering from an already challenging environment, might have been more capable of tolerating the cave's conditions than organisms adapted to pristine surface habitats. Third, the colonization may have been a gradual process, with organisms slowly expanding into the cave over many generations as evolution progressively adapted them to the changing conditions. Rather than a sudden, catastrophic isolation event, the sealing of the cave may have been gradual, the clay and less deposits accumulating slowly over thousands of years, progressively restricting the exchange between the cave and the surface, and gradually intensifying the selection pressure within the cave. In this scenario, evolution would have had time to keep pace with the changing conditions, adapting the organism step by step as the environment became more extreme. These possibilities are not mutually exclusive. The actual history of cave colonization likely involved elements of all three, with different lineages entering the cave at different times, with different degrees of pre-adaptation, and experiencing different rates of environmental change. Disentangling this history requires detailed molecular phylogenetic analysis, combined with geological reconstruction of the cave's environmental history, an ongoing research effort that is slowly but steadily filling in the details of this extraordinary evolutionary story. The question of how many species the cave actually harbors, and how many remain to be discovered, is another area of active inquiry. The figure of 50-plus species currently known from Movile Cave is almost certainly an underestimate. Microbial diversity in particular is likely to be far greater than currently documented, with many species detectable only through metagenomic approaches and many more potentially present in microhabitats that have not yet been sampled. Even among the macroscopic animals, new species continue to be described from the cave. Several of the cave species were not recognized as distinct from known cave or surface species until molecular analysis revealed genetic differences significant enough to warrant species-level distinction. Cryptic species that are morphologically similar but genetically distinct, are likely to be found when the cave's animal fauna is subjected to thorough molecular analysis. The microfauna, the community of organisms too small to see with the naked eye but large enough to be considered animals, including nematodes, rotifers, tardigrades, and various protozoa, has been particularly understudied in Movile Cave. These organisms occupy an important position in the food web, between the microbial mats and the larger invertebrates, and their diversity and ecology are almost entirely unknown. Future research using high-resolution sampling and advanced microscopy, combined with molecular taxonomy, is expected to reveal significant additional diversity in this size class. The cave's fungal community is similarly poorly known. Fungi are notoriously difficult to identify morphologically. Many species look similar and can only be distinguished by molecular analysis or chemical profiling. Metagenomic studies of the cave's fungal community are expected to reveal a diversity of species comparable to or exceeding that of the bacteria, with many novel taxa adapted specifically to the cave environment. The prospect of continued discovery in Movile Cave, of new species, new metabolic pathways, new ecological relationships, is one of the most exciting aspects of ongoing research. Despite 4 decades of study since its discovery, the cave remains far from fully characterized. Each new study, employing ever more sensitive and sophisticated techniques, reveals additional layers of biological complexity. The cave is not a simple system that has been fully described. It is a complex world whose full inventory and understanding may take another century or more to complete. Let us pause and reflect on the singular achievement of the organisms of Movile Cave, not as passive products of their environment, but as active agents that have solved, over millions of years, an extraordinary set of engineering problems. Consider what these organisms have achieved. They have built a complete, self-sustaining ecosystem in a sealed underground chamber, without any input from the outside world beyond the chemical energy of the deep earth. They have developed biochemical machinery to extract energy from chemicals that are poisonous to most life. They have maintained stable populations in a tiny, resource-limited space for millions of generations. They have evolved sophisticated sensory systems that allow effective navigation, prey detection, and mate finding in conditions of absolute darkness. They have developed tolerance to atmospheric conditions, low oxygen, high sulfide, high carbon dioxide, that would kill most surface organisms in hours. And they have maintained all of this in stable operation, through ice ages and warm periods, through the rise and fall of civilizations, through the entire history of the human species, without any assistance, without any input, and without any awareness of the world above. This is an extraordinary accomplishment, not conscious, not intentional, but no less remarkable for that. Evolution is a blind engineer, selecting solutions to problems by the simple criterion of which organisms survive long enough to reproduce. It has no foresight, no planning, no goal. And yet, over millions of years, this blind process has produced organisms of astonishing sophistication, organisms that make a living in conditions that seem designed to prevent any living thing from surviving. The lesson is humbling. It tells us that life's ingenuity, the range of solutions it can find to the challenge of surviving in a hostile world, is far greater than we might imagine. Every time we have thought we understood the limits of life, that we had identified conditions too extreme for any organism to tolerate, we have been proven wrong. Hot springs too acidic for life, wrong, there are bacteria thriving in pH 0 environments. Too cold, wrong, there are organisms in Antarctic ice living at temperatures well below freezing. Too salty, wrong, there are halophiles in salt lakes so saline that the salt precipitates on the lake floor. Too dark, wrong, there is Movile Cave. Too toxic, wrong, there is Movile Cave. Life is not fragile. It is not confined to a narrow range of comfortable conditions. It is an extraordinarily robust phenomenon, one that once established has the capacity to adapt to almost any stable set of conditions, however extreme, given enough time and a sufficient supply of energy. The timeline of life in Movile Cave, 5.5 million years, invites us to consider the deeper history of chemosynthesis on Earth. If the cave's ecosystem has persisted for 5.5 million years, unchanged in its fundamental character while human civilizations have risen and fallen above it, what does this tell us about the longevity of chemosynthetic life in other contexts? The deep subsurface of the Earth, the rock of the continental crust, kilometers below the surface, harbors microbial life. This has been established by drilling projects that have recovered live bacteria from depths of several kilometers, from rocks that have been isolated from the surface for millions to hundreds of millions of years. These deep subsurface organisms live at extraordinarily slow metabolic rates, sustained by trace amounts of chemical energy, hydrogen produced by the reaction of water with iron-bearing minerals, or the decay of radioactive elements in the surrounding rock. Some estimates suggest that the total biomass of life in the deep subsurface may be comparable to the total biomass of life on the surface, a startling claim that would, if true, mean that half of all life on Earth lives below the surface, in conditions of darkness, limited energy, and geological timescales of activity. The deep subsurface biosphere would then not be a marginal phenomenon, a thin biological film on the edge of the habitable zone, but a major component of Earth's living systems, operating on a different timescale and by different energetic rules than the surface biosphere, but no less real and no less significant. The organisms in the deep subsurface may reproduce on timescales of hundreds or thousands of years, their entire life cycle from birth to reproduction to death, playing out over a span that would encompass all of recorded human history. They are not just slow by the standards of surface biology, they are operating on a fundamentally different relationship with time. For them, a million years is not a geological epoch, it is a few thousand generations. It is a period during which evolution can make significant changes to the genome, adapt the organism to shifting conditions, and drive speciation. The deep subsurface biosphere, like Movile Cave, operates on the slow rhythm of the geological world rather than the rapid pulse of the biological surface world. And it reminds us that time is perhaps life's greatest resource, that given enough time, even the most extreme environment can be colonized, the most hostile chemistry can be harnessed, and the most improbable adaptations can evolve. Movile Cave gives us a glimpse of this slow, deep geological biology, not in the inaccessible depths of kilometers of rock, but in a space small enough to crawl through, at a depth shallow enough to reach by hand, in conditions extreme enough to be revealing but not so extreme that we cannot study them. It is the closest thing we have to a window into the deep subsurface world, a compressed, accessible, studyable version of the hidden half of Earth's biosphere. As the research on Movile Cave continues, international collaboration has become increasingly important. Romanian scientists who discovered the cave and have conducted the most sustained research program have been joined by researchers from universities and research institutions across Europe and increasingly from other continents. This internationalization of Movile Cave research reflects both the growing global interest in the cave and the need for diverse scientific expertise: microbiology, molecular biology, ecology, evolutionary biology, geology, geochemistry, astrobiology, that no single institution can provide. Collaborative research programs have produced some of the most significant advances in our understanding of the cave. Joint Romanian-German-British expeditions in the 1990s established the basic ecological parameters of the cave, the species list, the trophic structure, the atmospheric and water chemistry. Later international collaborations produced the first molecular phylogenetic analyses of cave organisms, the first metagenomic studies of the microbial community, and the first detailed isotopic characterizations of the cave's food web. Future collaborative projects are expected to focus on whole genome sequencing of cave organisms, detailed functional genomic studies of adaptation mechanisms, and long-term monitoring programs to track changes in the cave's ecosystem over time. The application of new technologies, autonomous sampling robots, advanced chemical sensor arrays, next-generation sequencing platforms, promises to dramatically accelerate the pace of discovery in the coming decades. There's also growing international interest in the cave from the perspective of astrobiology and space exploration. Space agencies and research programs interested in the exploration of icy moons have recognized Movile Cave as a valuable terrestrial analog, a place where the techniques and strategies for detecting chemosynthetic life in a sealed subsurface environment can be tested and refined before being deployed in a planetary mission. Collaborations between cave researchers and space scientists are producing new analytical tools, new detection strategies, and new frameworks for thinking about what biosignatures, chemical or physical signs of life, might look like in a chemosynthetic subsurface environment. There is one more dimension of Movile Cave that deserves acknowledgement before we bring our exploration to a close, the dimension of wonder. Pure, unalloyed wonder at the existence of a place that should not exist, populated by creatures that should not be able to live, sustaining itself by means that we had not imagined, for timescales that dwarf all of human history. Wonder is not a scientific measurement. It cannot be quantified or published in a peer-reviewed journal. But it is arguably the most important product of discovery, the emotional and intellectual response that drives scientists to ask the next question, to push the next limit, to look in the next dark corner. The discovery of Movile Cave provoked wonder in everyone who learned about it, wonder at the strangeness of the cave's chemistry, wonder at the blindness and pallor of its inhabitants, wonder at the sheer improbability of a complete ecosystem operating without sunlight in a sealed chamber that no human had ever entered. That wonder is productive. It drives research. It attracts scientists to the field. It inspires young students to pursue biology, chemistry, geology, and astrobiology. And it keeps alive the sense that the natural world is far stranger, far more complex, and far more rich with possibility than any textbook can contain. Movile Cave is a reminder that surprises remain, that even in an age of satellites and particle accelerators and genome sequencing, nature can still reveal something genuinely, profoundly unexpected. Consider the last paragraph on the story. 5.5 million years ago, as the clay and less accumulated above the cave, cutting it off from the sunlit world, the organisms inside had no choice. They could not escape, they could not fly to a new habitat, they could not migrate to better conditions. They were trapped, sealed in with the sulfide and the darkness and the thin air, with no sunlight and no future by any reasonable estimation. And they survived. Not just survived, they thrived. They adapted. They diversified. They built an ecosystem. They evolved spiders and leeches and water scorpions and crustaceans of extraordinary sophistication. They developed sensory systems and detoxification pathways and metabolic strategies tailored to the precise conditions of their prison. They turned a death sentence into a home. If that is not a statement about the endurance and creativity of life, about its refusal to accept limits, its insistence on finding a way wherever a way can be found, then nothing is. The sealed world beneath the Romanian plains continues its ancient clockwork even as we speak. The thermal waters rise from the deep earth, carrying their cargo of hydrogen sulfide and methane. The bacteria at the air-water interface receive these chemicals and do what they have done for millions of years: oxidize them, extract energy from them, convert them into the organic carbon that feeds the food web. The amphipods graze, the leeches hunt, the spiders listen. The pseudoscorpions creep through their miniature shadows. Above the cave, the sun rises and sets, seasons change, centuries pass, civilizations are born and crumble. The surface world is in constant, restless motion, changing, adapting, sometimes collapsing, sometimes flourishing. And beneath it all, Movile Cave ticks along at its own pace, slower, quieter, darker, stranger than anything on the surface, but alive. Stubbornly, triumphantly, incomprehensibly alive. This is the story of Movile Cave. It is a story about what life can do in the dark. It is a story about evolution's patience and creativity. It is a story about the hidden world beneath the world we think we know. It is a story about the boundaries of the possible and how, again and again, life pushes past those boundaries into territory that we had declared off limits. And it is, ultimately, a story about hope. If life can build a world in a sealed chamber without sunlight, powered by nothing but the Earth's internal chemistry, operating for 5.5 million years in conditions that we find toxic and unlivable, then life is more tenacious, more resourceful, and more widespread in the universe than even the most optimistic of us had dared to imagine. Somewhere in the cold, dark ocean beneath the ice of Europa, or in the hydrogen-rich waters of Enceladus's interior, or in some environment we have not yet thought to look, life may be doing exactly what it does in Movile Cave. Quietly, patiently, invisibly in the dark, oxidizing its chemicals, grazing its microbial mats, evolving, enduring, waiting perhaps for the day when another curious, wondering species punches through the roof of its world and shines a light inside. The cave will keep its secrets for a while longer, but each year, each study, each careful expedition brings us closer to understanding what it is telling us about the nature of life, about the history of our planet, and about the possibility that in the universe's vast, dark spaces, life is less alone than we have feared. The darkness of Movile Cave is not empty. It never was.